Immunology, the first topic is the lymphoid structures. Immune system organs. So there are two types of immune system organs. The first is primary organs and the second is secondary organs. So primary organs includes bone marrow and thymus. The function of bone marrow is the immune cell production and B cell maturation. So B cells get produced in bone marrow and it matures in the bone marrow itself. Whereas the function of thymus is T cell maturation. T cells are produced in bone marrow, but then they are matured in thymus. And secondary organs include spleen, lymph nodes, tonsils, and Peyer's patches. And they allow immune cells to interact with the antigen. Right. Spleen, lymph nodes, tonsils, and Peyer's patches. So first is lymph node. It is a secondary lymphoid organ that has many afferents and one or more efferents. It is encapsulated, this is important, with trabeculi A and B that we'll see later on. The functions are non-specific filtration by macrophages, circulation of B and T cells, and immune response activation. So the function of lymph node is the non-specific filtration. It has three parts: follicle, medulla, and paracortex. Follicle is located in the outer cortex. It is the site of B cell localization and proliferation. So the primary follicles are dense and quiescent, so they are inactive, and secondary follicles have pale central germinal centers and they are active. Right. So there are primary follicles and secondary follicles. This is the primary follicle and secondary follicle. So primary follicles have inactive B cells and active follicles are, as you can see, are large. They have pale central area that is called germinal center, and they are active. Okay. And the medulla consists of so the follicles are in the outer area of the lymph node, right? So these are the follicles. So as you can see, it is in the outer area of the lymph nodes, and the medulla are on the inner side of the lymph nodes. Medulla consists of medullary cords first and medullary sinuses second. So the medullary cords contain closely packed lymphocytes and plasma cells, and medullary sinuses contains reticular cells and macrophages. And medullary sinuses communicate with the efferent lymphatics, which is the exiting lymphatics. Here are the medullary cords, as you can see. Right. This is the medullary cord. Okay. This diagram doesn't show medullary sinuses. Okay, and paracortex, it is the area between the between the follicle and the medulla. It contains T cells. So it is the region of cortex between follicles and medulla. It contains high endothelial venules through which T and B cells enter from blood. It is underdeveloped in patients with Dijod syndrome. Okay, and paracortex enlarges in extreme cellular immune response, that is in EBV and other viral infections, which will cause paracortical hyperplasia, which is going to cause lymphadenopathy. So any lymphadenopathy we see in any infectious diseases, that is due to paracortex hypertrophy. So if the paracortex is hypertrophy, it is going to form more T cells to fight the infection, right? Okay, and in Dijod syndrome, there is the paracortex area is underdeveloped, so there is less T cells. Okay, so there we can also see thymic aplasia. So there is absence of thymus shadow in Dijod syndrome. And it contains high endothelial venules, so it has gaps, right? Endothelial venules are just gaps through which T and B cells enter from blood into the lymph nodes. Right. So this is the efferent lymphatics. So first there's follicles, then there's primary and secondary follicle. The primary is quiescent, secondary is active follicle with pale central germinal center. Then the secondary follicle also has a marginal zone and mental zone. Right? The outer zone is called mental zone. Right? And then there's paracortex, as we talked about, between follicles and the cortex area. So, and after paracortex there is medullary cords and medullary sinuses. So medullary sinuses communicate with the efferent lymphatics, and the follicle together contains is in the cortex area. The follicles are in the cortex area. Then this is the paracortex area, right? And then there's the medullary area. Okay. So this is the post capillary venule. This is capillary supply. Right. So that the blood is entering from the arteries. So there are these are the endothelial venules through which B and T cells enter the lymph nodes. These are the trabeculi, the capsule. As we talked about, the lymph node is a encapsulated lymphoid organ, and this is the medullary sinus. So see, medullary sinus is communicating with the efferent lymphatics. So now we'll see the lymphatic drainage associations quickly. So submandibular and submental area. Area of body drained is oral cavity, anterior tongue and lower lip. The malignancy of and metastasis to the oral cavity. Okay, so every malignancy related to the oral cavity is drained by submandibular and submental lymph nodes. Then there's deep cervical lymph nodes, which drains head, neck and oropharynx. So in upper respiratory tract infections, infectious mononucleosis, Kawasaki, malignancy of head, neck and oropharynx, not oral cavity, they are drained by deep cervical nodes. Then the supraclavicular node, so it is right is draining from right hemithorax, and left is also draining from left hemithorax as well as abdomen and pelvis. This is clinically very relevant, as in gastric cancer we saw that lymphatic metastases are can be found in the supraclavicular lymph nodes, right? That is called Virchow's node. And if it's in the left axillary lymph node, then it is Irish node, yeah. And malignancy of thorax, abdomen and pelvis, okay, these are the associated pathologies. Then there's mediastinal. Mediastinal is in the center, and hilar is in hilars are lateral to the mediastinal lymph nodes. Mediastinal drains from trachea and esophagus, because trachea is in the midline and esophagus is also in the midline, somewhat in the midline, right? You can remember it that way. And hilar drains from the lungs, as hilar lymph nodes are lateral to the mediastinal and lungs are also lateral. You can remember it that way, or you can just remember hilar with lungs. That is very simple. And the associated pathologies are pulmonary TB. There is just unilateral hilar lymphadenopathy, and in sarcoidosis there will be bilateral hilar lymphadenopathy, and TB is a caseous granulomatous disease, whereas sarcoidosis is non-caseous granuloma. And then lung cancer and granulomatous diseases. Then axillary lymph nodes, it drains upper lymph, breast, skin above the umbilicus, right? Skin above the umbilicus. And the associated pathologies are mastitis and metastasis, especially the breast cancer. Then epitrochlear, it drains hand and forearm, and it is associated with secondary syphilis. This is important too. So this is very specific to secondary syphilis, this epitrochlear lymphadenopathy. Then the celiac lymph nodes, it drains liver, stomach, spleen, pancreas and upper duodenum, till upper duodenum, everything. And superior mesenteric from lower duodenum to splenic flexure. Okay. And inferior mesenteric from splenic flexure to upper rectum. Okay. So they are associated with mesenteric lymphadenitis, inflammatory bowel disease and celiac disease. And between the superior and inferior mesenteric lymph nodes, there is para-aortic lymph nodes. So the name itself says that If this is the aorta and this is the parasite, this is the para-aortic lymph nodes. Right, so para-aortic lymph nodes, you can remember it with pair of organs, like pair of testes, pair of ovaries, pair of kidneys, and pair of fallopian tubes, and also uterus. And periumbilical, that is sister Mary Joseph node, it drains abdomen and pelvis, and it is associated with gastric cancer. Right, so many lymph nodes are associated with gastric cancer, as we saw. The first is paraumbilical, then there was left axillary lymphadenopathy, then the supraclavicular lymph nodes, all of them. And then there's external iliac, and there's internal iliac. The external iliac is in the lateral side of, is on the lateral side of the left side of iliac artery. Okay, so that is external iliac, and the internal iliac is in the medial side of iliac artery. So external iliac drains cervix, vagina, the upper third of the vagina, superior bladder, and body of uterus. Whereas internal iliac drains lower rectum to anal canal above the pectinate line, and bladder, vagina, the middle third of the vagina, cervix, and prostate. So the upper 1/3 of the vagina is drained by external iliac, and the lower 1/3 of the vagina is drained by internal iliac. Okay, and also the rectum above, the upper rectum is drained by inferior mesenteric lymph nodes, right? From splenic flexure to upper rectum, it is drained by inferior mesenteric, and from lower rectum to anal canal, just above the pectinate line, it is supplied by internal iliac, and below the pectinate line, it is supplied by superficial inguinal. Okay, yeah, and so superficial inguinal, it drains anal canal below the pectinate line, skin below umbilicus except popliteal area, then scrotum, vulva, and vagina, the lower third. So the upper third of vagina is supplied by external iliac, middle third by internal iliac, and lower third by superficial inguinal. And also anal canal below the pectinate line, and skin below the umbilicus. So, and the skin above umbilicus is drained by the axillary lymph node. Right? Here we saw axillary drains skin above the umbilicus, and superficial inguinal drains skin below the umbilicus. They are associated, the external, internal, and superficial inguinal, all three of them are associated with sexually transmitted infections, medial foot, leg cellulitis, and superficial inguinal. So the leg cellulitis is associated with superficial inguinal nodes. And the last is popliteal. So that drains dorsolateral foot and posterior calf. So that will be associated with lateral foot leg cellulitis. Okay, because it drains the dorsolateral foot. And the medial foot cellulitis, it is associated with these superficial inguinal. Okay. And all the solid markings of lymph nodes in this diagram they are the palpable lymph nodes, and all the ring-like lymph nodes, the empty lymph nodes, they are non-palpable lymph nodes. So which are the non-palpable lymph nodes? This is the mediastinal, hilar, this is superior mesenteric, then this is celiac, then superior mesenteric, then inferior mesenteric. The periumbilical lymph nodes are palpable, then para-aortic, then external and internal iliac. These are the non-palpable lymph nodes. All other are palpable lymph nodes. Yeah, right lymphatic drains right side of the body above the diagram into junction of the right subclavian and internal jugular vein. Okay, right lymphatic duct drains right side of the body above the diaphragm. So this is the right lymphatic duct circulation. Okay, right side of the body above diagram into junction of right subclavian and internal jugular. So right subclavian somewhere over here, and internal jugular vein. Okay, so The right lymphatic duct drains into the junction of both of them. Now the spleen. Okay. So first of all, I would like to mention that spleen does not have endothelial venules, or there is no T and B cell specificity like in lymph nodes. So let's just write that down. It is located in the left upper quadrant of abdomen, anterolateral to the left kidney, protected by 9th and 11th ribs. So whenever there is a case of RTA or trauma stating that the lower ribs of left sides are damaged, you can suspect splenic laceration or splenic injury. Okay. And so splenic dysfunction, there is post splenectomy or sickle cell disease autosplenectomy. The splenic dysfunction will lead to decreased IgM and decreased complement activation. So there will be decreased C3b opsonization by C3b. So that will lead to increased susceptibility to encapsulated organisms. So which are the encapsulated organisms? They are strep pneumo, which will cause sepsis. Then there's Haemophilus influenza, and there's Neisseria meningitis, malaria, babesia, all of them. So post-splenectomy findings are Howell-Jolly bodies, that are the nuclear remnants. Then there's target cells, there's thrombocytosis, because if there's splenic dysfunction, there's after post-splenectomy, the spleen will not be able to sequestrate all of the extra platelets, right? And also there will be lymphocytosis for the same reason. So let's see the images of Howell-Jolly bodies. So it is a basophilic substance in RBCs, which shows us the nuclear remnants or the DNA remnants. Then there are target cells, and the Howell-Jolly bodies and target cells, they are both seen in beta thalassemia as well. Okay, the vaccinate patients undergoing splenectomy or with splenic dysfunction against encapsulated organisms that will cause pneumococci, HIV, Haemophilus influenza type B, and meningococci. Because as we saw, these are strep pneumo, H influenza, and Neisseria meningitis. They are the encapsulated organisms which has increased susceptibility after in post splenectomy patients. Let's see the now let's see the parts of the spleen. The first is periarteriolar lymphatic sheath. So where's that? Yeah, here. So this is the periarteriolar lymphoid sheath. If this is the artery, so it is periarteriolar, which means surrounding the arteries. This is the lymphoid sheath. And if it is the lymphoid sheath, it contains T cells. And as it is lymphoid sheath, it is located within the white pulp. Then there's follicle. Okay, follicle contains B cells, just like lymph nodes. Here the follicle contains B cells. It is located within the white pulp. And then there's marginal zone. There's the marginal zone. Yeah. So the outer zone is the marginal zone. and the inner zone is the mental zone, and the central pale area is called the germinal center. So marginal zone contains macrophages and specialized B cells, site where antigen presenting cells capture the blood-borne antigens for recognition by lymphocytes. It is located between red pulp and white pulp. And ironically, in this diagram, the darker area, the pink looking area, is the white pulp, and this pale area is the red pulp. Okay, this is the red pulp, and the darker area is the white pulp, and and between them lies the marginal zone, right? So the marginal zone is the site for the antigen presenting cells to capture the antigens of the blood and present it to the lymphocytes. We'll see what antigen presenting cells are. They can be macrophages, they can be dendritic cells or natural killer cells or anything. So the red pulp is here. Okay, and red pulp has reticular fibrous framework, and this is the spleen has open circulation. And so this is the open circulation as well as closed circulation. Closed circulation leads in the sinusoids. So these are blood-filled sinusoids, and this is the open circulation. Okay. And in venous congestion, like in congestive heart failure and all that, or in liver cirrhosis, if there is venous congestion, there is red pulp expansion. Because there is open circulation, so if the blood is being congested in the body, blood can get accumulated through this open circulation into the red pulp, which will going to cause red pulp expansion. Okay, now thymus. Okay, and one more thing. We talked about the splenectomy. We talked about the splenic dysfunction. In splenic dysfunction, there is going to be decreased marginal zone macrophages, right? So marginal zones, as we talked about, contains macrophages and specialized B cells, right? So that is the most important part of the spleen. So if there is a splenectomy, there is going to be decreased marginal zone macrophages. And we saw that there is reduced C3b. So C3b is responsible for the opsonizations of the encapsulated bacteria, right? So that is the reason encapsulated bacterial susceptibility increases, because the encapsulated bacteria are mainly removed by the C3b opsonization. Okay. So now thymus is located in the antero-superior mediastinum. It is the site of T cell differentiation and maturation. So B cells maturation and differentiation both occurs in the bone marrow itself, but T cell maturation occurs in the thymus. Okay. It is encapsulated just like lymph nodes. Thymus epithelium is derived from the third pharyngeal pouch, and that is endoderm, whereas thymic lymphocytes are of mesodermal origin. Right? And the cortex is dense with immature T cells, and medulla is pale with mature T cells. Okay, so the cortex is dense with immature T cells, and when it matures, it is in the medulla. And Hassel's corpuscles containing the epithelial reticular cells. Now let's compare these site and the cell with those of the lymph nodes. So in lymph nodes, and this is the thymus. So the T cell, we have B cells and we have T cells. So in lymph nodes, the B cells were formed and proliferated in the follicles, and there is no B cells in thymus. And the T cells and lymph nodes, they are in the paracortex area, the area between the follicle and the medulla, and T cells in thymus are in the cortex as well as medulla. In cortex it is in immature state, and in medulla it is in mature state. Okay, let's talk about spleen as well. In spleen, the B cells are in follicles, just like lymph nodes, and the T cells are in periarteriolar lymphatic sheath. And if we talk about macrophages too, in lymph nodes, the macrophages are in medullary sinuses. So in medullary sinuses, there are macrophages as well as reticular cells, and in medullary cords, there were lymphocytes and plasma cells. Right, cords, lymphocytes, and plasma cells. And medullary sinuses contain reticular cells and macrophages. In thymus, there are no macrophages. And in the spleen, the macrophages and specialized B cells are in the marginal zone. Okay. Yeah, let's move forward. So the normal neonatal thymus is cell shaped on chest X-ray. So this is the thymus. Okay. It involutes by age 3 years, so we can't make out this shadow after 3 years. The T cells contain thymus, B cell contain bone marrow. Not contain, but bone marrow contains B cell, thymus contains T cells. Absent thymic shadow or hypoplastic thymus seen in immunodeficiencies like in SCID or Dye-George syndrome. We'll see the immunodeficiencies that causes T cell dysfunction or T cell deficiency. So SCID is and Dye-George are one of them. And thymoma, that is neoplasm of thymus, it is associated with myasthenia gravis, superior vena cava syndrome, and pure red cell aplasia and Good syndrome. So myasthenia gravis is an autoimmune disease, right? So antibodies are directed towards the postsynaptic acetylcholine channels. So those antibodies or those are secreted by the T cells that are from the thymus. So that thymoma can result in myasthenia gravis. Okay, and if there is thymoma, there is enlargement of the thymus gland. It can also compress the superior vena cava, which is causing superior vena cava syndrome. So there will be congestion in both arms as well as head in superior vena cava syndrome. Now the cellular components of immunology, the innate versus the adaptive immunity. So innate immunity components are neutrophils, macrophages, monocytes, dendritic cells, natural killer cells. There are those of lymphoid origin, complements, physical epithelial barriers, and secreted enzymes. Whereas adaptive immunity, the immunity which the body has adapted to fight a specific antigen that includes T cells, B cells, and circulating antibodies. Right, these are all specific immunities. Right, so circulating antibodies to specific antigens, or B cells or T cells secreted after released after uh, the infection by a specific pathogen. Right, those are all adaptive immunity. So mechanism of innate immunity is germline encoded. It is from birth. And the adaptive immunity, it is variation through VDJ recombination, that is the I think light chain or the heavy chain. Let me just they have a heavy chain. So variation of the heavy chain during lymphocyte development. Okay, so the recombination takes place according to the antigen it is targeting. Right? And the response to pathogens, it is non-specific, it occurs rapidly, and it has no memory responses. And response to pathogens in adaptive immunity is it is highly specific, it is refined over time, and develops over long periods. Then the memory response is faster and more robust. So innate immunity is there is no memory response, it occurs very rapidly, but it is a weak immune response. Whereas the adaptive immunity, it takes long period to develop, but it is a strong immune response. And the secreted proteins in innate immunity, it is lysozymes, complements, C-reactive proteins, defensins, and cytokines. Whereas in adaptive immunity, it is immunoglobulins and cytokines. Okay. And the key features in pathogen recognition is the toll-like receptors, uh, pattern recognition receptors that recognizes the pathogen-associated molecular patterns, that is PAMPs, and lead to activation of NFK beta, that is the transcription factor. And examples of PAMPs, so that is lipopolysaccharide in gram-negative bacteria, flagellin in red bacteria, and nucleic acids in viruses. So what happens is, so these are the pathogen associated molecular patterns, right? These are all the patterns. So if the pathogens has LPS or flagellins and or nucleic acids of viruses, they act as the molecular patterns that will be recognized by the toll-like receptors, right? And the toll-like receptors will activate the innate immunity. And these toll-like receptors are present on macrophages, dendritic cells, natural killer cells, all of those cells which are involved in the innate immunity. Right? So the other PAMs are like endotoxin, that also has lipopolysaccharides. Then there's peptidoglycan cell wall, that is in the bacteria. And then there's mannose, that is in yeast and bacteria and in lactin pathway. And there's also lipoteichoic acid, double strand RNA, or unmethylated DNA. They all act as PAMs that will act on toll-like receptors. And in adaptive immunity, there are memory cells, activated B and T cells, subsequent exposure to a previously encountered antigen, which will cause stronger and quicker immune response. Okay. So the key feature in adaptive immunity is the memory cells. So the second attack against the antigen is going to be more stronger and quicker. The immune privilege. The organs like eye, brain, placenta, and testis, and tissues where suppressive mechanisms limit immune response to foreign antigens to avoid damage that would occur from inflammatory sequel. So allograft rejection at these cells are less likely. Okay. So eye, brain, placenta, and testis, they have reduced immune responses, right? eyes, because every day dust goes into your eyes, and if the eyes were very sensitive towards the foreign antigens, then there would be inflammatory reactions every day, right? So eye has suppressed immunity, brain has suppressed immunity, placenta, because it is like placenta is it's going to be in contact with developing fetus, right? So it has to suppress the immunity to in order to decrease the inflammation due to any antigens, right? And testis. Allograft rejection at these sites are less likely. Okay. Now, MHC encoded by HLA genes. So MHC is the major histocompatibility complexes. It is 1 and 2. And MHCs are present on the exterior surface of the cell membrane, right? This is the cell membrane, this is on the extracellular space, and it presents antigen fragments to T cells and bind T cells receptors. It presents antigen fragments, not the whole antigen. So antigen is previously converted into antigen fragments, and then those antigen fragments are presented to the T cells, okay, and bind the T cell receptors, that are TCRs. So there are two types of histocompatibility complex, MHC1 and MHC2. So MHC1 contains HLA-B, HLA-A, B and C. They all have one letters. So MHC1. And MHC2 contains HLA-DP, DQ and DR. They both have, all three of them have two letters. Okay. So MHC2. Now the binding is to the T cell receptors and CD8 cells, and MHC2 binds to T cell receptors and CD4. Okay, we'll see the rule of 8, then you'll easily remember the CD8 and CD4 part. So the structure contains one long chain and one short chain in MHC1. So this is the long chain and this is the short chain. And in MHC2 there are two equal chains, two alpha chains and two beta chains, alpha 1, alpha 2 and beta 1, beta 2. So this is the one chain, this is the another chain. So they both are equal chains. And expression is all nucleated cells, antigen presenting cells and platelets except the RBCs. Okay, so all of these cells express MHC1 complex. Okay, and antigen presenting cells contains MHC2. APCs can express both MHC1 as well as MHC2. Right? But platelets and nucleated cells like WBC, they all express MHC1. The functions is it presents endogenous antigens like viral or cytosolic proteins to CD8 plus cytotoxic T cells. Cytosolic proteins, the cytosolic proteins can be of pathogens as well as from our own body cells. Okay. And function of MHC2 is present exogenous antigens, that is bacterial proteins to CD4 helper T cells. It is bacterial proteins. And here it was cytosolic proteins, which can be of virus or can be of our own body cell. So antigen loading, antigen peptides loaded onto MHC1 in rough endoplasmic reticulum after delivery via transporter associated with antigen processing, TAP. So after the antigen is processed into antigen peptides, it is transported to the MHC1 in rough endoplasmic reticulum, and antigen loaded following release of invariant chain in acidified endosome. So MHC1 is functioning in rough endoplasmic reticulum and MHC2 is functioning in acidified endosomes. And associated proteins are beta 2 microglobulin in MHC1 and invariant chain in MHC2. So like there's the rule of 8. So the answer should always be 8, 4 into 2 or 1 into 8. Right. So what is 4 into 2 and 1 into 8? So if the MHC is 1, the CD type of cell, it should be 8. So that the answer is 8. And if the MHC is 2, the CD cell should be CD4 plus. So the answer is 8. Okay, that's how you can remember. And so the long chain, the MHC1 has one long chain and one short chain, and the one long chain is the alpha chain. So that has alpha 1, alpha 2 and alpha 3 parts, and the alpha 3 parts goes inside the cell as well. And the short chain is the beta 2 microglobulin. Okay. Sorry. Now the HLA subtypes associated with the diseases, these are very important. And we'll also add some other HLAs which are not given here. So the first, let's see those that are given here. So the B27 is associated with PAIR, that is psoriasis, ankylosing spondylitis, IBD associated arthritis, that is inflammatory bowel disease, and R, that is reactive arthritis or Ritter's syndrome. Okay, can't see, can't pee, can't climb a tree. Okay, and B57 is associated with abacavir hypersensitivity, it is low yield. And DQ2 and DQ8, it is associated with celiac disease. This is again very important. So I ate too much gluten at the Dairy Queen. Okay, 8 for the HLA DQ8 and 2 for the DQ2 at Dairy Queen, that is HLA DQ2 and HLA DQ8. Dairy is for the gluten is for the celiac disease. DR3, that is DM type 1, SLE, Graves' disease, Hashimoto's thyroiditis, and Addison's disease. Okay, so DM type 1, you can remember it by HLA 3 and 4. How's that? Okay, yeah, so DM type 1 is also associated with DR4, okay, and DR3. So that's how it is, 4 minus 3 type 1. Okay, and SLE, so E is symbolized by 3. Then there's Graves' disease, Hashimoto's thyroiditis, and Addison's disease, basically all autoimmune diseases. And DR4 is associated with rheumatoid arthritis. So one room has 4 walls, so DR4, DM type 1, and Addison's disease. Now we'll see the other HLA subtypes. So A3 is associated with hemochromatosis. B8 by myasthenia gravis, Addison's, and again Graves. Then DR2, it is by multiple sclerosis, hay fever, SLE, and DR5 by pernicious anemia. Right. You can take a moment to memorize these, because I am taking one. Okay, now the functions of natural killer cells. So it is the natural killer cells are lymphocyte members of innate immune system, right? It uses perforin granzymes to induce apoptosis of viral infected cells, not viral but virally infected cells and tumor cells. Right. Activity enhanced by IL-2, IL-12, interferon alpha and interferon beta. We'll see those when we'll see the cytokines, that IL-2 and IL-12 are responsible for natural killer cells activity. It is induced to kill when exposed to a non-specific activation signal on target cell or to an absence of inhibitory signal such as MHC1 on target cell surface. So natural killer cells kills whenever there is non-specificity. If there is specificity towards one cell, if it is a viral cell, it is going to activate MHC1 and then cytotoxic T cell, or if there is bacterial proteins, it is going to activate MHC2 and then helper CD4 cells. If there is no specific activation signal on target cell, or there is inhibitory signal, absence of inhibitory signal such as MHC1 on target cell surface, so the natural killer cells come into defense. So the name itself suggests that it is a natural killer cells. So it is going to kill. It is its natural response, right? It also kills via antibody dependent cell mediated cytotoxicity. The CD16 binds the Fc region of bound IgG, activating the NK cells. And there's CD56 on surface of NK cells, which is found in neuromuscular junctions. And as it uses perforins and granzymes, they are the preformed enzymes, right? So there is the immune response, which is very quick and weak, like an innate immune system. Now the major functions of B and T cells. So the B cells is for humoral immunity and the T cells are for cell mediated immunity, because there are two types of cells, CD4 and CD8 T cells. So that causes cell mediated immunity. In B cells it is humoral immunity. The functions of B cells are these three. The first is recognize and present the antigen, second is produce the antibody, and third is maintain immunologic memory. So the first function, recognize and present the antigen, it undergoes somatic hypermutation to optimize antigen specificity. So as it is specified to a certain antigen, it is going to recognize it correctly and it is going to then present the antigen. Produce antibody, that is differentiate into plasma cells to secrete specific immunoglobulins and maintain immunologic memory. So memory B cells persist and accelerate future response to antigens. So once the B cells are activated, it can differentiate into plasma cells or memory B cells. If it is the plasma cells are going to secrete specific immunoglobulins, whereas the memory cells are going to persist and accelerate future response to the antigens. Now the T cells, it causes cell mediated immunity. So the CD4 plus T cells help B cells make antibodies and produce cytokines to recruit phagocytes and activate other leukocytes. Right. So it helps B cells to make antibodies, and it produces cytokines to recruit phagocytes and activate other leukocytes, which cytokines that we'll see later on. Okay. So the helper T cells are differentiated into Th1 and Th2. So if it is Th1, it is going to secrete interferon gamma, and if it is Th2, it is going to secrete interleukin 4, 5, 10 and 13. 4 is going to then stimulate phagocytes and other leukocytes, that is B cells. It promotes growth of B cells, and promotes secretion of IgE and IgG, and B, if the interleukin 5 is secreted, it is going to promote the growth of B cells and IgA and also eosinophils, that is the leukocytes, as we saw. Right, it produces cytokines through Th1 and Th2 cells. It recruits phagocytes and activates other leukocytes. As we saw, the IL-4 was activating the B cells, and the IL-5 was going to secrete eosinophils as well as the B cells. Right. And the CD8 plus T cells directly kill virus infected and tumor cells via perforin and granzymes. So there's no bullshit. It directly kills, right? It directly kills the virus infected cells or tumor cells via perforins and granzymes, similar to NK cells, as we saw here. The endogenous antigens, that is viral or our own cytosolic proteins, they are presented to the CD8 plus cytotoxic T cells. So it is going to directly kill them. Right? That is why they are called the cytotoxic T cells. They are toxic to all kinds of cell. Delayed cell mediated hypersensitivity, type 4. The T cells are also involved in type 4. There is no antibody involvement in type 4. Antibodies are involved in type 1, 2 and 3, but not in type 4. In type 4 there's just T cells, and there's acute and chronic cellular organ rejection. Okay, right. So right. So in acute and chronic cellular organ rejection, there is involvement of T cells, but not in hyperacute and graft versus host disease, because in hyperacute there is involvement of preformed antibodies that are always in the recipient. Okay, so the preformed antibodies in the recipient attacks on the donor antigens, but in acute and chronic cellular organ rejection there is involvement of T cells. It can be donor T cells or it can be recipient's T cells. Okay, that we'll see when we discuss the transplant rejections. Okay. And yeah, here's the rule of 8 that we talked about. MHC2 into CD4 that equals to 8, and MHC1 into CD8 that equals to 8. Okay, now let's briefly discuss the migration of neutrophils towards the inflammation or infection site, right? So the first stage is the rolling of neutrophils. So neutrophils rolls and then binds to the endothelium of the blood vessel. First, the neutrophils are in the center of the blood flow. This is the blood vessel. First, initially the neutrophils are here. Then during inflammation, the blood flow is slowed down and the neutrophils are moved towards the periphery. Okay, and then the neutrophils start rolling on the endothelium and binds to the endothelium. It binds E-selectin or P-selectin. The second stage is the crawling. So now the neutrophils secrete integrins. And those integrins binds to the endothelial cell. And the cells which it binds to are called ICAM cells. The third is transmigration. So it has rolled over to the endothelium, bound to the endothelium. It started crawling. It started secreting integrins that binds to the ICAM cells on the endothelium, and now there is transmigration. That is, the neutrophils migrate through the vessel wall. So now neutrophil binds PCAM1 between the endothelium cells. On endothelial, not on, but between the endothelium cells. Okay. And then this migration. Once it has transmigrated, once it has crossed the vessel wall, it now migrates to the inflammation site. Pardon my handwriting, guys, please. I know they are bad, and it's like handwriting of a 5-year-old, but I can't help it. Okay. It migrates to the site of inflammation, and then it secretes the chemokines C5a, that is complement C5a, and interleukin 8. So interleukin 8 is responsible for the migration of neutrophils to the site of inflammation. That we'll see when we discuss the different cytokines. Okay. And one more thing, the IL-1 and TNF alpha, they activate the selectins. So IL-1, IL-6 and TNF alpha are responsible for the acute inflammation. So that is how IL-1 and TNF alpha plays the role in acute inflammation, by activating the selectins. And LPS, lipopolysaccharides, and C5a stimulates the integrins that is secreted from the neutrophils. As differentiation of T cells, so the first is it is secreted from the bone marrow, right? The T cell precursors are secreted from the bone marrow, and it is differentiated in the thymus, as we saw earlier. Okay. So T cell precursors travel to thymus. In thymus, it matures, and it has the property to form both CD8 plus cells or CD4 plus cells. Okay, so CD8 plus and CD8 plus, they have receptors of both. They have these are the T cell receptors. Okay, so this is the T cell receptor of CD8 cells, this is the T cell receptor of CD4 cells, and this is the T cell receptor for MHC. For now it is MHC1 or MHC2. Okay. So at this point there is positive selection. Okay. So what is positive selection? That is occurring in the cortex of the thymus. Cortex is the outer part, right? So the positive selection occurs in the cortex part, and the negative selection occurs in the medulla part. So what is positive selection? It is the double positive CD4 plus and CD8 plus. Yes, we saw that. T cells expressing T cell receptors capable of binding self MHC on cortical epithelial cells survive. Okay, so the cells which are capable of binding the major histocompatibility complex, they survive. Okay. Let me just take out my own notes. I have written it in a more concise way. Okay, so the positive and negative selection occurring in the thymus is a part of central tolerance. Okay, so to prevent autoimmunity in our body, we have two kinds of tolerance, that is central tolerance and the peripheral tolerance. So central tolerance is occurring in the thymus. So what's happening here is that so the cells that are able to recognize the antigens plus the MHC complex, only those cells are going to survive. So the positive selection is not very complex as I thought. That is the negative selection, that is the complex part. So simply the positive selection allows the T cells that are able to recognize MHC complex. You just have to remember this. Only those cells are going to survive. So that is the positive selection. Now the negative selection, that is in the inner part, that is in medullary part. Okay, that is the negative selection. So, and the T cells expressing T cell receptors with high affinity for self antigens, that can cause autoimmunity, undergoes apoptosis or become regulatory T cells. Tissue restricted self antigens are expressed in the thymus due to the action of autoimmune regulator gene. So this is the AIRE gene. Okay, and the deficiency of that leads to autoimmune polyendocrine system 1. Okay, well, we'll see that later on. Okay. So in negative selection, if the lymphocyte is found to bind to the self antigens, it undergoes apoptosis, right? Because in order to prevent the autoimmunity, because if we allow the cell that can bound to the self antigens of the body, that can lead to autoimmunity in future, right? So we have to kill those cells which are binding to the self antigens. Okay, and how it is done, what is the mechanism of checking that if lymphocytes are binding to the self antigens or not? It is checked by exposing them to the dendritic cells or the medullary epithelial cells, okay, by the action of the AIR transcription factor gene. Okay, so let me write it down. So the AIR gene, okay, exposes the double positive cells to dendritic or medullary epithelial cells. Okay, and those cells that does not those cells that binds to these dendritic and medullary epithelial cells, they are killed. They will undergo apoptosis, because medullary epithelial cells are our own cells, right? It is the epithelial cells that are found in the medullary part of the thymus. Okay, so that are our own cells. So if the lymphocytes are acting towards those our own cells, then it has to undergo apoptosis. So those lymphocytes that binds to them, they are going to apoptosized. Okay, and what if this gene is mutated? What if the AIR gene is mutated? So then we will not have any check mechanism on these lymphocytes that can bind to the self antigens. So those lymphocytes will escape this negative selection and they will be secreted into the circulation. So that will have the ability to bind the self antigen, and that will lead to autoimmune syndrome, that is autoimmune polyendocrine syndrome 1. So autoimmune polyendocrine system syndrome 1, it includes chronic mucocutaneous candidiasis, hypoparathyroidism, adrenal insufficiency, and recurrent candida infections. So that is CAR. Candida, chronic candida, hypoparathyroidism, adrenal insufficiency, and recurrent candida infections. So this was central tolerance in thymus, but what about the central tolerance in bone marrow? So let's write somewhere over here that in bone marrow there are stem cells, right? Hematopoietic stem cells that turns into immature cells and eventually into naive B cells that are released in the circulation that will mature in the lymph nodes. Okay. So there is negative selection over here. We are talking about central tolerance. We will also talk about the peripheral tolerance, but for now the central tolerance in bone marrow. So how does this negative selection takes place? So the cells will be the naive B cells or the immature B cells will get exposed to the dendritic cells to see if they bind to it very tightly. And if they are binding very tightly to the dendritic cells, there will be expression of the RAG genes, the RAG genes, and there will be editing of the light chains of the immunoglobulin. So when we edit the light chains of the immunoglobulin, it will no longer bind to the self antigens, or it will no longer bind too tightly to the dendritic cells. So that was the first option, the editing of the light chains of immunoglobulin, and the second option is directly apoptosis of that immature B cells which were binding too tightly to the dendritic cells. Okay, that's it. That's the central tolerance in bone marrow. So if they bind too tightly to dendritic cells, one of the two things can occur. The first is expression of RAG genes. So that will cause editing of light chains of immunoglobulin. And second is apoptosis. So that is the central tolerance. And if even then somehow the cells, these cells that binds to the self antigen escapes the central tolerance, there is always peripheral tolerance, right? So let's discuss the peripheral tolerance. So also in peripheral tolerance, two things are happening. Okay, so if these self-reactive CD4 lymphocytes are binding to self antigens, right? So if they are binding to the self antigens, there wouldn't be any secondary co-stimulatory signal. We'll see what the secondary co-stimulatory signals is a little later on, but for now just remember that there is co-stimulation that is called a secondary signal. So if that is absent in the self-reactive CD4 lymphocytes, it will go energy. Okay, and the second point is it can also undergo apoptosis just like in central tolerance, but here it is not. Here it is due to Fas ligand binding to the Fas receptor. Okay, so self-reactive lymphocytes binding to antigen without co-stimulatory signal. They are going to undergo energy. And second, Fas ligand can bind to Fas receptor on the cell membrane, so that will stimulate apoptosis. And also there is autoimmune lymphoproliferative syndrome, okay, that is ALPS. Autoimmune lymphoproliferative syndrome. So there is mutation in the Fas ligand apoptotic pathway. Mutation in Fas pathway. So that would not result in the apoptosis of those lymphocytes. So there will be lymphoid accumulation that is going to cause lymphadenopathy and lymphoma in future. Lymphadenopathy and lymphomas. And there are also regulatory T cells, okay, to prevent autoimmunity. So how does regulatory T cells function? So regulatory T cells are CD4 plus cells. Regulatory T cells develop from the CD4 plus type of T cells, okay, and there is CTLA4 on T regulatory cells that is going to bind B27 receptor on B cell, and once it binds to the B27 receptor of the B cell, it secretes IL-10, TGF beta, okay, and they both attenuates the inflammatory response, right? So if even there is autoimmune reaction, it is going to suppress that, okay. Okay, now let's see the development of the differentiation of the T cells. Okay, so we saw there's T cell precursor, T cell precursor, which gives rise to double positive CD4 CD8 T cells. It can either form CD8 plus T cell, or it can form CD4 plus T cell. CD8 plus T cell is definitely going to form cytotoxic T cell, right? So cytotoxic T cell will have CD8 plus receptor and T cell receptor, which will bind MHC1. So that is MHC1. Okay. And CD4 plus T cell first gives rise to the helper T cells, and helper T cells can gives rise to different kinds of Th cells like Th1, Th2, Th17, or T regulatory cells. Okay. So first is Th1 cells. So interferon gamma and IL-12. Okay. So IL-12 is important over here. Interferon gamma and IL-12 helps helper T cells to form Th1 cells, and Th1 cells are secreted in the peripheral blood, and it secretes interferon gamma and IL-2. And what is the function of IL-2? IL-2 activates macrophages and cytotoxic T cells. Okay. Now helper T cells with help of interleukin 2 and interleukin 4 forms the Th2. Okay. And Th2 secretes IL-4, 5 and 6 that we'll see later on, and 4, 5, 6 together activates eosinophils, right? Like IL-5 is responsible for activation of eosinophils, and IL-4 is responsible for increase in IgE. Okay, and it also secretes IL-10 and IL-13. IL-10 attenuates the inflammatory response, so it suppresses the inflammatory reaction. So it prevents the chronic, it prevents any inflammation in or infection going into the chronic stage. Okay, it stops it at the acute stage. And helper T cell with the help of TGF beta, IL-1 and IL-6, those are the acute phase reactants, all three of them, forms Th17. So Th17 is going to secrete IL-17, 21 and 22, and it will induce neutrophilic inflammation. As we said that it is acute phase reactants, so it is going to cause inflammation, and for inflammation neutrophils are required. And now helper T cells with the help of TGF beta and IL-2 gives rise to regulatory T cells. Okay, and regulatory T cells secretes TGF beta, IL-10 and IL-35, that is going to prevent autoimmunity, as we just saw. Okay. Okay, so Th1 is for macrophage and cytotoxic T cells, Th2 is for eosinophils and IgE, Th17 is for neutrophilic inflammation, and T regulatory cells are for preventing the autoimmunity. Okay, so now macrophage lymphocyte interaction. So Th1 cells secrete interferon gamma, which enhances the ability of monocytes and macrophages to kill microbes they ingest. Okay, this function is also enhanced by interaction of T cell CD40 ligand with CD40 receptor on macrophage, and macrophage also activate lymphocytes via antigen presentation. Okay, so Th1 cells will secrete interferon gamma, which is going to activate macrophages to kill the microbes they just ingested. Okay, and in return macrophages also activate lymphocytes via antigen presentation. Okay, so in short, if this is the macrophage and this is the T cell, so macrophage is first going to present the antigen to the T cell. So in response of the antigen presented to the T cell, Th1 cell is going to secrete interferon gamma, that is going to activate macrophage, that is going to kill that antigen. Okay, so that is this cycle. We'll also see this later on in detail. Okay. And cytotoxic T cell, it kills virus infected, neoplastic and donor graft cells by inducing apoptosis. Okay, that is cytotoxic T cell. It is toxic to any kind of cell infected by virus, either neoplastic or virally infected or donor graft cells. Okay, or cytotoxic proteins as well. It releases cytotoxic granules containing preformed proteins, that is perforin and granzyme B. Cytotoxic T cells have CD8, which binds to MHC1 on virus infected cells. Okay, the regulatory T cells help maintain specific immune tolerance by suppressing CD4 plus and CD8 plus T cell effector functions. It is identified by expression of CD3, CD4, CD25 and FOXP3. Okay. And CD5 is associated with multiple sclerosis and DM type 1, and FOXP3 is associated with IPEX syndrome. And activated regulatory T cells produce anti-inflammatory cytokines that are IL-10 and TGF beta. So that halts the acute inflammation to its acute stage. It does not allow the acute inflammation to go into the chronic stage. Okay. Now this is important that it is expressed by CD3, CD4, CD25 and FOXP3. Okay, and now the IPEX syndrome. So IPEX is I for immune dysregulation, P for polyendocrinopathy, E for enteropathy, and X for X-linked. It is the genetic deficiency of FOXP3. So there is going to be autoimmunity. It is characterized by enteropathy, endocrinopathy, nail dystrophy, dermatitis, and other autoimmune dermatological conditions. It is associated with diabetes in male infants. That is part of the polyendocrinopathy. Now the T and B cell activation. So as I earlier said, that antigen presenting cell can be anything. It can be B cell, it can be dendritic cell, Langerhans cell, or even macrophages. Two signals are required for the T cell activation, B cell activation, and class switching. Okay. So first we'll see the T cell activation. So there is antigen presenting cell. This is the antigen presenting cell which will receive the antigen. It will ingest the antigen and it will the antigen is going to travel to the MHC2 or MHC1 part. Okay, it is not specific. It is at this point of time MHC1 or MHC2, and it is going to present the antigen to the T cell receptor on the naive T cell. So once antigen is received on the T cell receptor, there is going to be another signal as well. As we talked about earlier, there is going to be a secondary signal, that is the co-stimulatory signal. So what is the co-stimulatory signal? The B7 of the antigen presenting cell binds to the CD28 of the naive T cell. So once that happens, the first signal is this one, the antigen binding to the T cell receptor, and the second signal is the B7 binding to the CD28 of the naive T cell. Once both of these signals are stimulated, there is going to be activation of the naive T cells. Okay, and once the T cell is activated from its naive stage, it is going to produce cytokines. Okay, and those cytokines are going to kill the virally infected cells. Okay. Now let's see the theory part, that APC ingests and processes the antigen, then migrates to the draining lymph node. So we know that lymph node is the site for the antigen presenting cell to present antigen to the T lymphocytes. Okay. So second is T cell activation. So signal 1 includes exogenous antigen is presented on MHC2 and recognized by T cell receptor on THCD4 cells. Endogenous and cross-presented antigen is presented on MHC1 to TC cytotoxic T cell CD8 plus cells. So if it is exogenous antigen, it is going to be presented via MHC2, and if it is endogenous antigen or cross-presented antigen or self-antigen or cytosolic proteins, it is going to be presented on MHC1. And this was the first signal. And what is the second signal? The second signal is the proliferation and survival. Okay. So it is the co-stimulatory signal via interaction of B7 protein, that is CD80 and CD86 on dendritic cells, and CD28 on the naive T cell. Okay. So these are the two signals. So the first is T cell activation and the second is proliferation and survival. And the activated TH cell produces cytokines, and the cytotoxic T cell able to recognize and kill virally infected cells. So if it is the helper T cell, the process does not stop here. It's going to produce cytokines. But if it is a cytotoxic T cell, TC cell, the process is going to stop there by T cell recognizing the pathogen and kill the pathogen, right? Cytotoxic T cell directly kills the virus infected cells. It does not need any cytokines. And as we talked about, antigen presenting cell can be any cell as long as they have major histocompatibility complex MHC1 or MHC2 that can present the antigen to activate the naive T cells. Okay. Okay, and once the CD4 plus cells are activated through MHC2, the CD4 plus T cells are activated. Then the CD4 plus T cells are going to convert it into either Th1 or Th2 cells that are going to release the interleukins. Okay, so that interleukins are going to form the inflammatory response against the pathogen. Or if it is MHC1, it is going to activate CD8 T cell that is directly going to kill. By the preformed proteins that are perforins and granzymes. Now as the T cells are activated, we'll see how the B cells get activated through the T cells. Okay, so in first case we saw the antigen presenting cells presenting the antigen to the naive T cells. Now we'll see how the B cells, so B cells can also activate as the antigen presenting cells, right here it is written that antigen presenting cell can be a B cell too. So now if B cell is presenting the antigen through the MHC2 to the activated T cell through the T cell receptor, right, and there is a secondary signal as well here too, that the CD40 of B cell binds to the CD40 ligand of the T cell. So that's how the T cell CD4 plus T cell gets activated, and it is going to produce cytokines that is going to activate the B cells. And as the B cells are being activated by the cytokines, the B cells undergoes in immunoglobulin class switching. So initially the immunoglobulins are just in the form of IgM, but it has to undergo class switching to produce different different immunoglobulins like IgA, IgE, IgG. So that is called the class switching of the immunoglobulins. So for the class switching to occur, we need cytokines from the activated CD4 plus T cells. Okay. So the T cell activation as above, right? So T cell activation is this part, that antigen is binding to the B cell receptor. So antigen is then being transported to the lymph node and presented via MHC2 to the T cell receptor, and the T cell is being activated. TH helper T cell is being activated, and it is if it is helper T cell, it is CD4 plus T cell. And then B cell receptor mediated endocytosis. Okay. No, sorry, the first stage is TH cell activation as above. So via this mechanism, the T cell has activated. Okay. Let's forget about what I said, that antigen goes into the B cell. That is not the first step. The first step is the activated T cell. From the T cell activation comes here. So the second stage is B cell receptor mediated endocytosis. So this is the B cell receptor mediated endocytosis. So the antigen gets endocytosed into the B cell, and then the exogenous antigen is presented on MHC2 and recognized by T cell receptor on TH cells. So the antigen is going to MHC2 and will bind to the T cell receptor on helper T cells. And fourth is CD40 receptor on B cell binds to CD40 ligand on helper T cells. Okay. and the helper T cells secrete cytokines that determine Ig class switching of B cells. So it secreted cytokines that are going to determine the Ig class switching of B cells. So now B cells are again activated and produce IgM, and they undergo class switching and affinity maturation. Okay, so what's happening is APC with its antigen via MHC1 or 2 is binding to the T cell receptor on T cell along with B7 receptor of the antigen presenting cell binding to CD28 of T cell. Okay, so that activates the T cell okay, so that can form either a cytotoxic cell or it can form helper T cell, right? And if it is undergoing helper T cell, it is going to produce cytokines that kills this is the activated TH cells. So now B cell can also act as antigen presenting cell with its antigen. Okay. So that binds to the activated TH cell via MHC2 to the T cell receptor of the helper T cells. So when the B cell is binding to the TH cell and also the CD40 of B cells binding to CD40 ligand of the helper T cells, it is going to make activated TH cell produce cytokines and those cytokines are going to activate Ig class switching. And affinity maturation. Okay. Also, how does this cytokine, what is the mechanism of producing the cytokines? Okay, let's see the mechanism, that how does the activated helper T cell produce the cytokines. So once antigen is presented to the T cell receptor, it is going to activate IKB kinase, and activation of IKB kinase is going to, it is IKB kinase, right? So it is going to cause ubiquitination of the IKB and once the IKB is destroyed, it is ubiquitinated. There is going to be release of NFK beta. It is the transcription factor, and this NFK beta is going to enter the nucleus and promote synthesis of cytokines, acute phase reactants, and sialization molecules. And CAMs, sialization molecules. And so NFK beta is bound to the IKB in cytoplasm in inactive stage. So once the IKB is inactivated or ubiquitinated, so NFK beta is now free. So that free NK beta can travel up to nucleus and act as a transcription factor. Because NFK beta and IKB are bound together in the cytoplasm. Yeah, and also another clinical correlation is that this, as we talked about, the CD40 of the B cell binds to the CD40 ligand of the T cell. So if this stage is defective, it is going to cause hyper IgM syndrome. Because if this stage is defective, there is no going to be binding of CD40 to CD40 ligand, and there is not going to be any production of cytokines, and there is not going to be any immunoglobulin class switching in the B cells. So that is going to cause deficiency of all immunoglobulins, but just increase in immunoglobulin M or IgM, because initially just IgM is formed, and it is after the immunoglobulin class switching that other immunoglobulins are formed, like IgA, IgE, and IgG. Okay, so there is going to be increase in IgM, and there is going to be reduce in Ig, A, G, and E. Okay. So now we'll see the immune responses. So the first topic is antibody structure and function. So the Fab fragments consist of light and heavy chains that recognizes the antigens. Okay. So which one is the Fab region? Yeah, this is the Fab region that gets exposed to the antigens and it recognizes the antigens. And FC region of the IgM and IgG fixes complement. Okay, of IgM and IgG fixes complements, and so this is the FC part. So that is for complement binding. Okay, and heavy chain contributes to FC and Fab regions both. So if this is the heavy chain, so this is the Fab region, and this is the FC region. It is contributing to the both of them, and light chains contribute only to the Fab region. This is the light chain, it is contributing just to the Fab region. Okay, so Fab is fragment antigen binding. So that is the mnemonic antigen binding. Here is the antigen binding. Okay, determines idiotype, unique antigen binding pocket, only one antigen specificity expressed per B cell. Okay, so there is just one kind of antigen that can bind to one particular type of B cell, that is one antigenic specificity. And for FC region, we have to remember the 5 C's, that it is constant. It does not change like in the Fab region, that it changes per B cell. It is antigen specific. It is just one antigen specific, so it has to change every other cell. It is not like that. The FC region is constant. It has the carboxy terminal. It binds the complement. It has carbohydrate side chains, and it confers the isotype. Okay, that is it going to be IgM or IgD? So Fab region determines the idiotype and the FC region determines the isotype. And generation of antibody diversity, that is antigen independent. So the generation of antibody diversity and generation of antibody specificity. So first is how is antibody diversity generated? It is antigen independent. It does not depend on the kind of antigen it is binding to. So there is random recombination of VJ, that is light chains, or VDJ, that is heavy chains, genes by RAG1 and RAG2. So we also talked about the role of RAG genes in the central tolerance in the bone marrow, right? There is expression of RAG genes and there's editing of the light chains of immunoglobulin so that it does not no longer binds to the self antigens. Similarly, here the RAG1 and RAG2 genes are responsible for the recombination of the light chains or heavy chains, okay, so that the antibody can be diverse. It can bind diversity of antigens, number of antigens, okay. So there is random addition of nucleotides to DNA during recombination by terminal deoxynucleotidal transferase, that is TDT. Okay, so TDT adds nucleotides to the DNA. Okay, so that is called recombination. And the third is random combination of heavy chains with light chains. So all three of them results in antibody diversity. And generation of antibody specificity, it is antigen dependent. So there is let's say there is an antigen received. So now specific to that antigen, there is going to be somatic hypermutation and affinity maturation in the variable region. So which is the variable regions? This is the variable region, that this VL, variable region of light chain, variable region of heavy chain, and C is the constant of light, constant of heavy, okay. And here is the constant of heavy 2, constant of heavy 3. There is 3 constant fragments in the heavy chains, okay. And there are 2 fragments in the light chains. The one is light and one is variable and one is constant. And isotype switching, that is in the constant region, okay. So somatic hypermutation and affinity maturation, that occurs in the variable regions, and in the constant regions there is antibody specificity through isotype switching. Neutralization is antibody presents bacterial adherence, and once it adheres to the surface, there is opsonization. So antibody promotes phagocytosis, and then there's complement activation. So membrane attack complex, that is MAC, binds to the antigen surface, and C3b also binds to the antigen surface, which is going to cause opsonization. So antibody activates the complement, enhancing opsonization and lysis. So all isotypes can exist as monomers. So mature naive B cells prior to activation express IgM and IgD only on their surfaces. They may differentiate in germinal centers of the lymph nodes by isotype switching, that is gene rearrangement induced by cytokines and CD40 ligand. Remember the cytokines produced by the Th cells and CD40 of B cell binding to CD40 ligand of T cell. Right, here the cytokines produced by the T cells activating the B cells for Ig class switching, and CD40 ligand of T cell binding to CD40 of B cell promoting Ig class switching into. So this may differentiate in germinal centers of lymph node by isotype switching, gene rearrangement induced by cytokines and CD40 ligand into plasma cells that secrete IgA, IgG and IgE. For B cells, IgMOM and IgDad matures to plasma cells as they age. Affinity refers to the individual antigen-antibody interaction, while avidity describes the cumulative binding strength of all antibody-antigen interactions in a multivalent molecule. Okay. Affinity is for individual antigen-antibody interaction, and avidity is for cumulative binding strength of all antibody-antigen interactions. So IgG is the main antibody in secondary response to an antigen. It is the most abundant isotype in serum. Okay. It is the most abundant in serum, but it is the, if they ask which is the most produced antibody, it is IgA. Okay. So most produced antibody is IgA, but the most abundant is IgG, because IgA is produced in serum and then it leaves the serum and enters the GI tract, so its concentration relatively decreases in the serum. So IgG fixes complements, opsonizes bacteria, neutralizes bacterial toxins and virus. So basically it does everything that you expect from an immunoglobulin. So only isotype that crosses the placenta, it also crosses the placenta and provides infant with the passive immunity that starts to wane after birth. So IgG greets the growing fetus associated with warm autoimmune hemolytic anemia. And now let's first talk about IgM, then we'll go to IgA. Okay, so IgM is produced in the primary response to an antigen. It fixes complements just like IgG. It antigen receptor on the surface of B cell, monomer on B cell and pentamer with J chain when secreted. So when it is secreted, it is secreted in the form of a pentamer. So pentamer is binding of the 5 immunoglobulins, 5 IgM, and pentamer enables avid binding to the antigen while humoral response evolves, associated with cold autoimmune hemolytic anemia. Okay. So IgM is the primary response, that is the immediate response, and IgG is the secondary response. So that is why we say that IgM is responsible in the acute infection and IgG is raised in the chronic infection like in hepatitis, right? Because IgG is for the secondary response to an antigen. Now the IgA, it prevents attachment of bacteria and viruses to mucous membranes, does not fix complement. So that is the point that is different from IgM and IgG. It is monomer in circulation or dimer with J chain when secreted. Okay. It crosses epithelial cells by transcytosis, produced in GI tract in the Peyer's patches, that is the lymphoid aggregate in the submucosal layer, and protects against the gut infection, that is Giardia, and most produce antibody overall but has lower serum concentration. It is released into the secretions, tears, saliva and mucus and breast milk, and it picks up secretory components from epithelial cells, which protects the FC region, FC portion, from luminal proteases. Right. So what does it all means? It is produced in the GI tract, so it can provide GI immunity against the gut infections like giardiasis, and also to provide immunity in other secretions like tears, saliva, mucus and breast milk. It is abundant in those secretions, and it picks up the secretory component from epithelial cells, so which protects the FC region from the luminal protease. So there are protease enzymes, right, to digest food that we eat in the intestine. So that protease can also digest the immunoglobulin A, but in response to prevent itself, IgA binds to the secretory component of the epithelial cells, so the FC region can be protected against the proteases. Okay, now the IgD. There is unclear function of IgD found on the surface of many B cells and in serum. So if they say there is a B cell deficiency, there is also going to be IgD deficiency. Now IgE, the binding, it binds mast cells and basophils. It binds mast cells and basophils. Cross-links when exposed to allergen, mediating immediate type 1 hypersensitivity through release of inflammatory mediators such as histamine. It contributes to immunity to parasites by activating the eosinophils. Okay, it binds mast cells and basophils. It is responsible for the type 1 hypersensitivity reaction. Okay. Sorry. So when it binds to mast cells, there is a degranulation of those mast cells occurring, which is going to release different enzymes like histamine, tryptase, and leukotrienes and all that, which is going to mediate the type 1 hypersensitivity reaction, right? Now the antigen type and memory. So there can be two type of antigens. It can be either thymus independent antigen or thymus dependent antigen. So antigens that lack a peptide component that can stimulate an inflammatory reaction, or it can stimulate antibody secretion. So that peptide component, if the antigen is lacking that peptide component, or it is lacking that PAMs that we talked about, that peptide associated something like, 1 second, let me see that, like pathogen associated molecular pattern or something like that. Yeah, pathogen associated molecular patterns. If the antigen is lacking that, it is going to it cannot be then presented by MHC to the T cells, right? So there is weak immunogenic. So that is thymus independent antigens. So that why is it called thymus independent? Because as it lacks PAMs, as it lacks the peptide component that can lead to MHC binding to the T cells. So therefore the thymus is not getting activated. So that is why it is called thymus independent antigens. Antigens lacking a peptide component, it is lipopolysaccharides from gram-negative bacteria, or lipoteichoic acid, or mannose residues, or anything like that, or unmethylated DNA, or double-stranded RNA that we all talked about that cannot be presented by MHC to the T cells. So they are weakly immunogenic. Of course, because they are not being presented by MHC to T cells, there is no cytokine production, there is no B cell activation, there is no Ig class switching, there is no immunoglobulin secretions. So there is going to be less immune reaction, right? They are weakly immunogenic, and vaccines often requires boosters and adjuvants. So they are capsular polysaccharide subunit of Streptococcus pneumonia, that is PPSV 23, that we'll see when we discuss the vaccinations. Okay. And the thymus dependent antigens. So antigens containing a protein component like the diphtheria toxoid. Toxin is a protein component, right? So that is going to stimulate the class switching, and immunologic memory occurs as a result of direct contact of B cell with Th cells, helper T cells. So once the B cell is being activated via the helper T cells, it is going to secrete, it is going to undergo immunoglobulin class switching, and also undergo formation of memory B cells that is going to form an immunological response in future if the antigen is introduced once again. Right. So the B cell is binding to the Th cells. So helper T cells are from the thymus, right? they are proliferated and matured in thymus, right? So that is why it is called thymus dependent antigens. Okay, now the complements. So it is the system of hepatically synthesized plasma proteins, and it is plasma proteins that play a role in innate immunity and inflammation. So innate immunity was, as we discussed, it is the quick, weak response, right? And the adaptive response was a late response, but it was a stronger response via immunoglobulins. Okay. So complements play a role in innate immunity and inflammation, and membrane attack complex defends against gram-negative bacteria. The CH50 test is used to screen for activation of the classic complement pathway. So activation pathway. So classic can be IgG or IgM mediated. Alternative can be the microbe surface molecules, and lectin can be the mannose or other sugars on the microbe surface. Okay. We are going to see an easier way of remembering all these complement pathway. This might look very scary, but trust me, it's not. So we are going to discuss that in a easier way. So first, let's just go over the theory part. Like the C3b is responsible for the opsonization. You have to mug up this topic, that C3b is responsible for opsonization. Okay. Like C3b binds to lipopolysaccharides on bacteria. Okay, that is going to cause opsonization. C3a, 4a, 5a are for anaphylaxis. All the A's are for anaphylaxis, and C5a are also for neutrophil chemotaxis. Okay, and C5b to 9b, like 5b, 6b, 7b, 8b, 9b are part of the membrane attack complex that are responsible for cytolysis. And MAC complex is important for neutralizing the Neisseria species. So deficiency results in the recurrent infections of the Neisseria species. Okay. And which are the opsonins? As we first talked about, the C3b is responsible for opsonization, that is the thumb rule, and the other is IgG, are the two primary opsonins in bacterial defense. They enhance the phagocytosis. C3b also helps clear the immune complexes. Okay. And which are the inhibitors of the complement activation? Right, complements can't be good all the time. Like it can be bad if they are activated when you don't need them to get activated, right? So we need inhibitors that can inhibit the complement pathways from activating at the wrong times, right? So the first is decay accelerating factor. It is DAF, also called CD55, and C1 esterase inhibitor. It helps complement activation on the self cells. So if the complements are getting activated against the self antigens, if that's happening, so then these DAF and chlorine esterase inhibitors are going to inhibit those complements getting activated against the self antigens. And these are very important. DAF and C1 esterase inhibitors are very important, because if they are deficient, they are going to lead into two diseases that we'll discuss later on. Okay, now first let's just go over this pathway. So forget this diagram, we are going to draw our own diagram. So first, just write down the three pathways that are classical, alternate, and lectin pathway. The mnemonic is CAL. Okay, you can remember just CAL from California. So CAL. And now what you have to remember is 2, 3, 4. That is in the order, right? C2, C3, C4. So let's just write them down. C2, C3, C4. Okay. And whenever the complement is being activated, the activated form is of C2, C3, C4 is C2b, C3b and C4b. Okay, you have to remove the A component from the complement. Okay, now first let's discuss what activates this pathway, right? So classical pathway, as we saw, they are mediated by oh, they are mediated by IgM and IgG. Alternative pathway is by microbes, surface molecules, and lectin is activated by the mannose or other sugar on the surface of the microbes. But let's just remember the mannose or other sugars. Okay. Right. So now the C2, C3, C4 are going to get activated into C2b, C3b, C4b. Okay. And now let's just remember that C3b is the most important complement of all, right? Because it helps in opsonization. Okay, so just remember that C3b is the most important, and eventual step is to get everything to C3b, which will later on activate other complement like C5, C6, C7, C8, C9b. Okay, so now, as we said, C3b is the most important. So in the alternate pathways, C3b is formed directly, but in classical and lectin pathway, the C2b and C4b is formed, which has to convert into C3b. So that converts C3 to C3b. Okay, so all the pathway came to the C3b. Now what C3b does is, as I said, it activates the later pathways, like it is going to activate C5 to C5b. Right. And now C5b is going to convert into membrane attack complexes. Right. And what helps them does that the C6 to C9 helps C5b convert into membrane attack complex, which are responsible for the lysis, and cytotoxicity. Lysis and cytotoxicity. Okay, so this is the simplest as it can get. Okay, and as I said, the C3a part should be removed. The A part should be removed from all the complements to activate it into C3b. So here is C2a removed, here is C3a removed, here is C4a removed. Now let's just go over this diagram once. So like the classical pathway, so C2 is getting converted into C2b, and that is helped by C1. Okay, and C1 helps C2 get converted into C2b. C2a is removed, C2b is getting converted into C3 convertase. So here is the C3 convertase that is going to convert C3 into C3b if not already happened. Okay, and then alternate pathway, that is C3 getting converted into C3b via this BB that is activated by D. Okay, C3b is then activated into C3bb or something like that's funny. That is converted into C3 convertase, right? So C3 is converting into C3b. And in lectin pathway, there is C1 like complex. In classical, there was C1, but in lectin, there is C1 like complex that is going to convert C4 into C4b. C4a is removed. C4b is activates C3 convertase. So everything eventually comes down to C3 getting converted into C3b. Okay. So now as C3b is activated, it is going to activate C5b convertase here. So the name itself suggests that C5b convertase is going to convert C5 into C5b, and C5a is removed. So C5b is going to then turn into membrane attack complexes, that is C5b, C6b, C7b, C8b, C9b. These are called the terminal complements. Okay, from C5 to C9 they are terminal complements, and C3, C2, C3, C4 they are called early complements. Okay, and C5b is activated into MHC by C6 to C9. Okay, and MAC eventually causes lysis and cytotoxicity. Okay, just remember this Cal 2, 3, 4 and you are good to go, right? Now the complement disorders. So there can be complement deficiency or there can be regulatory protein deficiency. So they are complement regulatory protein deficiency, the DAF and the C1 esterase inhibitor that we talked about that inhibits the complement activation if they are being deficient. Okay. So first let's see the complement protein deficiency. There can be early complement deficiency, that is from C1 to C4, and terminal, that is C5 to C9. So if early complement deficiency is there is risk of severe recurrent pyogenic sinus and respiratory tract infections. Okay. C3b is used in clearance of antigen antibody complexes. So there can also be accumulation of antigen antibody complexes, and there is increased risk of SLE if there are nuclear antigens. Antibodies binding to the nuclear antigens will result in antigen antibody complexes in case of SLE, right? And terminal complement deficiency C5 to C9, as we talked about, that C5 to C9 are responsible for membrane attack complexes that are useful in getting rid of the Neisseria species infection. So if the C5 to C9 are deficient, recurrent Neisseria bacteremia can occur. It can be in form of meningitis or UTI, like in the form of gonorrhea. Okay. Neisseria meningitis, Neisseria gonorrhea. Okay. Or it can cause sepsis as well. The Waterhouse-Friderichsen syndrome or something like that. You have to look it up. And there will be going to be recurrent history of those infections. Right? There is not going to be any upper respiratory tract infection. Trust me, it is going to help you to differentiate different disorders. Like if it is upper respiratory tract infections, there can be deficiency of B cells. Okay, so in the cases of immunodeficiency syndromes, there will be going to be history of upper respiratory tract infections or GI tract infections. But in here there is no GI tract infections or upper respiratory tract infections. There is just meningitis, UTI or sepsis. All that can be caused by Neisseria species. And now let's just see C1 esterase inhibitor deficiency causes hereditary angioedema due to unregulated activation of kallikrein. Okay, so kallikrein, I guess, is stimulated by the complements. Okay, so now if there is a complement inhibitor deficiency, there is going to be lots of complement, and the lots of complements are going to activate kallikrein. That is going to cause increased bradykinin. Okay, it is characterized by decreased C4 levels. Okay. So why does it have decreased C4? That I don't even remember. Presence of non-functional. Okay. So as C1 esterase is deficient, the complement activation pathway is going to fire up. Okay, and once the complement activation fires up, as we saw in the lectin pathway, C4 is converted into C4b, and then it is converted into C3b, and then C5 and C9 and all that. So the initial stage is C4, right? So the C4 is going to decrease because all the C4 is being activated into C4b, which is used into activating C3 into C3b. So the result is C4 is going to be low in the cases of hereditary angioedema, right? And ACE inhibitors are contraindicated because they also increase the bradykinin, right? One of the key adverse effect of ACE inhibitors is increased bradykinin, causing cough and angioedema like symptoms. Okay. So the second is paroxysmal nocturnal hemoglobinuria. A defect in the PIGA gene prevents the formation of glycosyl phosphatidyl inositol GPI anchors for the complement inhibitors such as decay accelerating factor and membrane inhibitor of reactive lysis. Oh, that is lot of words. So DAF and CD59. Okay, this is CD55, and MIRL, that is membrane inhibitor of reactive lysis, is CD59. They both are the complement inhibitors. But they need GPI anchors. And for the formation of the GPI anchors, we need the PIGA gene. Okay, so now if the PIGA gene is mutated, there is not going to be formation of the GPI anchors, So now there will be no activation of the decay accelerating factor DAF or CD59 complement inhibitors. So now if there is no complement inhibition, there is going to be increased complements pathway. Okay, so that causes complement mediated intravascular hemolysis. So and whenever there is intravascular hemolysis, there is decreased haptoglobulin and dark urine. Okay, because all the heme is getting secreted into the cell. It is leaving the red blood cell. It is entering the circulation, causing dark urine. It can cause atypical venous thrombosis like in Budd-Chiari syndrome, portal vein, cerebral or dermal thrombosis. Also, I forgot to discuss one more thing with you, that early complement deficiency, mostly the C3 deficiency, causes pyogenic sinus and respiratory tract infections. Okay, so C3 deficiency mostly results in decreased opsonization, decreased opsonization causes susceptibility to encapsulated organisms. So which are the main encapsulated organisms? Right, the strep pneumo. Okay, so strep pneumo infection mostly causes upper respiratory tract infections. Okay, and sinus infections, mostly the pus forming type, that is pyogenic sinus infections. Right. And also, if you want to check for the hereditary angioedema, the best site to look for is the periorbital area. Okay. So you can make out angioedema easily in the periorbital area. And meningitis is always most commonly associated with terminal complement deficiency. If you see Neisseria mentioned anywhere in the questions, most probably it is going to be terminal complement deficiencies. Now is the most important page of this system. So first, let's see this in cytokines secreted by the macrophages. Okay, so macrophages secretes interleukin 1, interleukin 6, TNF alpha, interleukin 8 and interleukin 12. So first of all, the acute phase reactants, that are the first three, interleukin 1, 6 and TNF alpha, are secreted, and then they recruit IL-8 and IL-12. So for the neutrophilic chemotaxin, and the IL-12 for differentiation of T cell into the helper T cells. Okay. So whenever the macrophages phagocytose any kind of antigen, that is the initial response to phagocytose the antigen, then it is going to secrete interleukin 1, which is going to cause fever and acute inflammation. It also activates the endothelium to express adhesion molecules. So once adhesion molecules are expressed on the endothelium, neutrophils can, when the interleukin 8 is going to be secreted, it is going to attract the neutrophils. So if the interleukin 1 already activates the endothelium to express adhesion molecules, those neutrophils secreted through IL-8 are going to stick to the endothelium because of the adhesion molecules. Okay. It also induces chemokine secretion to recruit the WBC. See, it is also called osteoclast activating factor. So osteoclast activating, so osteoclast does the bone resorption, right, and demineralization. Okay. Interleukin 6 is responsible for fever, and it stimulates production of acute phase proteins. And acute phase proteins that are fibrinogen, ferritin, hepcidin, and C-reactive protein. They are all acute phase proteins that are increased in a inflammatory state. And then tumor necrosis factor alpha, it activates endothelium. It causes WBC recruitment and vascular leak. The clinical association of vascular leak is with DIC. Okay, so vascular leak occurs due to the TNF alpha in DIC. Okay, and it also causes cachexia in malignancy, that is tiredness and fatigue in malignancy, or the muscular wasting. Okay. It maintains the granuloma. Remind you, it does not form granuloma, but it maintains the granuloma. And IL-1, 6 and TNF alpha can mediate fever and sepsis. Okay. So how does it causes cachexia in malignancy? It is by inhibiting of lipoprotein lipase in fat tissues. And interleukin 8 is major chemotactic factor for neutrophils. Cleanup on IL-8. If this is a John Wick movie, there is a cleanup on IL-8. Okay, so there that's how you can remember it. Like after John Wick does its business round, he hires the team to clean up all the dead bodies from that area, from that aisle. So he has to call the interleukin 8 to clean up all the dead bodies. So in that sense, it has to recruit all the neutrophils to clean up the debris on the IL-8. Neutrophils are recruited by IL-8 to clear the infections. In interleukin 12, it induces differentiation of T cell into Th1 cell. Okay, so interleukin 12 helps us in going to the next step, that is activation of T cells into helper T1 cells. Okay, and it facilitates granuloma formation in TB. That we'll see how that macrophages activates. Macrophages release IL-12. IL-12 will then stimulate Th1. Th1 will stimulate interferon gamma. Interferon gamma will in turn activate macrophage to facilitate the granuloma formation and all that. We'll discuss this little later on. Okay, and it also activates natural killer cells. Okay, so this hot T-bone steak. Hot is for IL-1, T-bone for IL-2 for stimulating T cells, IL-3 for stimulating the bone marrow, and 4, 5, 6 for steak, EAK, IgE, IgA, and acute phase protein production. Okay, so let's just revise all of these. So interleukin 1 is for fever and acute inflammation and adhesion molecules and osteoclast activating factors. 6 is just for fever and acute phase proteins. TNF alpha is for activating the endothelium and recruitment of WBC and vascular leak, also maintaining the granuloma and cachexia in malignancy. Interleukin 8 for cleanup on IL-8, that is neutrophil activate chemotactic factor for neutrophils, which is going to clear out the infection. And interleukin 12 is for facilitating the granuloma formation in TB, as well as differentiation of T cells into Th1 cells, that is helper T cell 1. Okay. Now let's just first see Th1 cell. Okay, we'll see this interleukin 2 and 3 later on, but first we'll see Th1 cell so you can grasp the information better. So Th1 cells secrete interferon gamma, secreted by NK cells and T cells in response to antigen or IL-12 from macrophages. It stimulates macrophages. Remember I told you that in turn stimulates macrophages to kill the phagocytosed pathogen, right? So it stimulates macrophages to kill phagocytosed pathogen. It inhibits the differentiation of Th2 cells. Okay. Because if Th2 cells are activated, then there's going to be a chronic inflammation and activation of different immunoglobulins like IgE, IgG, IgA and all that. To prevent all of that inflammatory reaction and to hold the inflammation to just macrophages and Th1 cell, it inhibits the differentiation of Th2 cells. It induces IgG isotype switching in B cells. Okay. It increases major histocompatibility expression and antigen presentation by all cells. Okay. So if the question is what increases the MHC expression, it is due to interferon gamma. It activates macrophages to induce granuloma formation. Okay. So macrophages secrete IL-12. IL-12 stimulate the Th1 cell. It differentiates T cells into Th1 cell. Th1 cell is going to secrete interferon gamma. Interferon gamma in turn stimulates the macrophages to one, kill the pathogen, kill the phagocytosed pathogen, and second, to induce granuloma formation. Okay, so this is the cycle: macrophage, Th1 by IL-12, Th1 interferon alpha gamma activates macrophage. Okay, that is the cycle: macrophage, Th1 macrophage. Okay, now secreted by T cells. So before T cell differentiating into Th1 and Th2, it secretes interleukin 2 and interleukin 3. Okay. It stimulates, so interleukin 2 stimulates the growth of helper, cytotoxic and regulatory T cells and NK cells. Okay, obviously because it is secreted from the T cells before differentiating into Th1 and Th2. So if anything is secreted from T cells before it is differentiating, it is going to help with the differentiation, right? So interleukin 2 secreted from T cell before differentiation is going to help T cell differentiate into helper T cells or cytotoxic T cell or regulatory T cells and NK cells as well. And interleukin 3, it supports growth and differentiation of bone marrow stem cells. This is unique. It functions like granulocyte, monocyte, I think. I don't know. GM-CSF functions like GM-CSF. Okay. And interleukin 2, as it is stimulating the growth of all of these different kind of T cells, it should be suppressed, right, to block the immunity. Okay. Because if it is not suppressed, it is going to stimulate all kinds of cell, which in turn will stimulate different kinds of cytokines, going to secrete different kinds of cytokines, which is going to end up in a inflammatory response. So we don't need that at all times. So at all times, without any infection or any pathogen present, interleukin 2 should be suppressed. And also if it is being suppressed abnormally, even during the time of need, during the time of infection, if the IL-2 is suppressed, there is not going to be any immune response. So that can also cause immunodeficiency syndrome. If IL-2 can't act on the IL-2 receptor, it is going to cause immunodeficiency syndrome, that we'll see here. Here in the SCID, there is defective IL-2 receptor gamma chain, so interleukin 2 cannot act on the interleukin 2 receptor, so there is not going to be any kind of immune response. Right. Now the last Th2. So Th2 secretes 4 interleukins, that is 4, 5, 10 and 13. Interleukin 4, it induces differentiation of T cells into T helper cells. It promotes the growth of B cell and hence class switching to IgE and IgG to beg for help. Th2 BEG, that is B cells, immunoglobulin E and immunoglobulin G. Okay. And 4 help, that is through the interleukin 4, and help is helper T cell 2. You don't need to remember this, you can just remember 2 beg. So helper T cells promotes the growth of B cell and class switching of IgE and IgG. And interleukin 5 promotes growth and differentiation of B cells just like interleukin 4, but it enhances the class switching of IgA and stimulate and growth and differentiation of eosinophils as well. So 2 cells, B cells and eosinophil, and just 1 immunoglobulin, that is immunoglobulin A, that is 5B. So interleukin 5, B cell, IgA and eosinophil. So interleukin 4 does one more thing. Like interleukin 4 reduces the activity of Th1 cells and Th17 cells as well. And macrophages as well. So it is a kind of anti-inflammatory, because if you see here, during the T cell differentiation, interleukin 4, it inhibits the formation of Th1 cells and Th17 cells from the helper T cells. Okay. And it is promoting the differentiation of helper T cells into Th2 cells. As it is said here, induced differentiation of T cell into Th2 cells. Okay, and it enhances the class, interleukin 5 enhances the class switching to IgA. And as we all know that IgA is used against helminths and GI bacteria. Okay, so if there is interleukin 5 deficiency, there is going to be increased helminth and GI tract infections. Now the interleukin 10, it attenuates inflammatory response, decreases the expression of MHC class 2 and Th1 cytokines. Okay. And it inhibits activated macrophages and dendritic cells. It also secreted by the regulatory T cells. Obviously, the regulatory T cells will always want to reduce the inflammatory response, be it autoimmunity or even inflammatory response. So regulatory T cells will always want to decrease that by secreting the interleukin 10. Okay. So it is the expression of MHC class 2 and also Th1 cytokines. So if it is inhibiting Th1 cytokines as well, so that is interferon gamma, so there will be no granuloma formation, as interferon gamma is inhibited. Okay, and as there is no granuloma formation, there is no chronic inflammation, right? So it attenuates the immune response. It is therefore interleukin 10 and TGF beta. Both are anti-inflammatory reactions. Both are anti-inflammatory cytokines. Not TNF alpha but TGF beta. Okay, don't confuse them. Okay, now it also helps in healing and fibrosis. Okay. So it limits the acute inflammation. It does not allow the acute inflammation to go into the chronic inflammation stage. So it's a good interleukin, right? And in last is interleukin 13. It promotes IgE production. Just like interleukin 4, it promotes IgE production by B cells. It induces alternative macrophage activation. Okay, and interleukin 13 promotes IgE. So how is macrophage activated? It is activated by the interferon gamma secreted from the Th cells, right? But it is also alternatively it is also activated by the interleukin 13 formed from the Th2 cells. Okay. So interleukin 10 and TGF beta, they are going to prevent acute going into chronic, but all other cytokines from the Th2 they are going to form chronic inflammation. Because of all the immunoglobulin secretion, IgG is known for chronic inflammation, right? So because of all those interleukins secretion and immunoglobulin secretion, there is going to be chronic inflammation. Okay. Also one more point that IL-6 and TNF alpha, they are responsible for ubiquitin proteasome pathway for degrading the skeletal muscles and proteins. So that will lead to cachexia in malignancy. Like it stimulates pathway, ubiquitin proteasome pathway to degrade skeletal muscles and protein. Now the respiratory burst. It is also called oxidative burst. It involves the activation of phagocyte NADPH oxidase complex in neutrophils and monocytes, the cells which are able to phagocytose, and which utilizes O2 as substrate. It plays an important role in the immune response. Rapid release of reactive oxygen species. Okay. Immune response, rapid release of reactive oxygen species. Okay. It releases reactive oxygen species which are going to act on the bacteria and convert it into bacterial fragments. So NADPH plays a role in both the creation and neutralization of reactive oxygen species. And myeloperoxidase contains a blue-green heme-containing pigment that gives sputum its color. Blue-green color, it is due to the myeloperoxidase enzyme. So now, if this is neutrophil, this is the neutrophil cell membrane, and this is the bacterial receiving site. Okay, this is the bacteria. So what happens is oxygen is converted into oxygen free radical by NADPH. It is through the enzyme NADPH oxidase. So the name itself suggests that it is an oxidase enzyme. It is going to provide a extra proton ion, ion to the oxygen, converting into oxygen free radical with a negative charge. That is an extra electron giving it a negative charge. Then that free radical is converted into hydrogen peroxide, that is due to the enzyme superoxide dismutase. And hydrogen peroxide gets converted into HOCl through catalase or glutathione peroxidase. And that HOCl free radical is responsible for destroying the bacteria, for killing the bacteria into small fragments. As you can see, it ruptures the cell membrane or cell wall of the bacteria, okay, making it into fragments. Okay, and that H2O2 formed due to superoxide dismutase, that hydrogen peroxide also enters the neutrophil cytoplasm, and there eventually it helps in conversion of glucose 6-phosphate to 6-phosphogluconolactone, and mostly it is due to the NADPH. That is why in G6PD, as we talked about, there is not going to be G6 glucose 6-phosphate dehydrogenase, so that is going to lead to decrease in NADPH, and due to reduced NADPH, there is going to be lysis of the red blood cell, because there is not going to be enough glutathione reductase, right? Okay. And if there is a deficiency of the myeloperoxidase enzyme, like myeloperoxidase is used somewhere over here, and if it is deficient, there is going to be recurrent thrush infections, that is recurrent candida infections. And the nitroblue tetrazolium test is going to be normal, because nitroblue tetrazolium test is just used for the function of NADPH oxidase, but here NADPH oxidase function is normal. It's just that myeloperoxidase is deficient, so there is going to be normal nitroblue tetrazolium test as NADPH oxidase function is intact. And also selenium deficiency, the selenium compound selenium deficiency can lead to glutathione peroxidase enzyme deficiency as well. Where is glutathione peroxidase? Yeah, here. So selenium deficiency can lead to glutathione peroxidase enzyme deficiency, and B12 is required for glutathione reductase. So the phagocytes of patients with chronic granulomatous disease can utilize hydrogen peroxide generated by invading organism and convert it to reactive oxygen species. So patients are at higher risk of infection by catalase positive species, that is Staph aureus and Aspergillus, capable of neutralizing their own H2O2, leaving phagocytes without reactive oxygen species for fighting infections. So in chronic granulomatous disease, as it is a granulomatous disease, it is going to form hydrogen peroxide, right? So body can use that hydrogen peroxide released from those pathogens to convert it into reactive oxygen species, so that can help us making an immune response. But in infection by catalase positive species like in Staph aureus and Aspergillus, these catalase positive organisms, they have capability of neutralizing that H2O2 and neutralizing that H2O2 formed in the body so if the H2O2 is neutralized by these organisms, we will never have reactive oxygen species. We will never have HOCl, free radical or oxygen species for fighting infections, right? Because all of the H2O2 are being used up by the catalase positive organisms and neutralized by catalase positive organisms into H2O plus O2. So if there is no H2O2 for free radical formation of ROS formation, there is going to be no strength of fighting infection because of no ROS. And the pyocyanin of Pseudomonas aeruginosa generates reactive oxygen species to kill competing pathogens. It can generate its own ROS, and that will kill the competing pathogens. It will not going to kill Pseudomonas itself, but it is going to kill other competing pathogens. The oxidative burst leads to release of lysosomal enzymes. And lactoferrin is a protein found in secretory fluids and neutrophils that inhibits the microbial growth via iron chelation. That is a vague point. Lactoferrin is in the secretory fluids like in saliva, like in breast milk, and in neutrophils that will inhibit microbial growth via iron chelation. So now the interferons, there are three kinds of interferons that are used in the clinical practice or are produced in the body. It's interferon alpha, beta and gamma. So mechanism of interferons is, it is a part of innate host defense. Innate host defense, interferons interfere with both RNA and DNA viruses. So cells infected with virus synthesize these glycoproteins which act on local cells, priming them for viral defense by down-regulating the protein synthesis to resist potential viral replication and by upregulating MHC expression to facilitate recognition of infected cells. It also plays a major role in activating anti-tumor immunity. So it is an innate host defense, not an adaptive one. So innate host defense, so it is going to be very rapid and weak, and the interferons interfere with both RNA and DNA virus. So suppose if this cell is infected with RNA or DNA virus, so now what happens is these cells are going to secrete interferons, and suppose this is a normal cell, so these interferons are going to act on the normal cells and down regulate the protein synthesis and to resist potential viral replication and by upregulating MHC expression to facilitate recognition of infected cells. So these cells, these normal cells, can now recognize these virally infected cells. And also by secreting interferon, it is going to downregulate its own protein synthesis. So now if the protein synthesis is downregulated in this affected cell, so the viral replication is going to halt down. Okay. And its clinical uses in chronic HBV, Kaposi's sarcoma, hairy cell leukemia, condyloma acuminatum, renal cell carcinoma, malignant melanoma, multiple sclerosis, and chronic granulomatous disease. And the adverse effects are flu-like symptoms, depression, neutropenia, myopathy, and interferon-induced autoimmunity. Neutropenia, myopathy, interferon-induced autoimmunity. Cell surface proteins. So cell surface proteins of T cells include the T cell receptor CD3 and CD28. Okay. So TCR, the T cell receptor, binds antigen of the antigen MHC complex. Okay. MHC complex brings the antigen to the T cell receptor. CD3, it is associated with T cell receptor for signal transduction. And CD28 binds the B7 on antigen presenting cell. Okay, the B7 is on antigen presenting cell and CD28 is on T cells. Now for the specific helper T cells, there's CD4, as it is a helper T cell, it is going to have CD4. Then the CD4 ligand, which binds to CD40 of B cells, right? And there is CXCR4 and CCR5. They are associated with pathogenesis of HIV. And cytotoxic T cells just have CD8, right, because it is cytotoxic, which is going to bind to MHC1. Okay. And regulatory T cells have CD4 and CD25. As we talked about, the regulatory T cells have CD4, CD25, and FOXP3. And the mutation of FOXP3 was causing IPEX syndrome, right, where there is autoimmunity. No. Okay, so now B cells. Sorry. So the B cells have immunoglobulins that binds the antigen. So immunoglobulin is a receptor after all, right? That is going to bind the antigen. In addition, it has CD19, 20, 21. They are the receptor for Epstein-Barr virus, and CD40, that is going to bind CD40 ligand of the T cell, helper T cell, and there's MHC2 and also B7. And these CD19, CD20, CD21, you have to remember for the B cell lymphomas too, right? Because they are the cytokines that are being expressed on the B cell lymphoma. Okay, and NK cells just have CD16 and CD56. So CD56 is, as we talked about earlier, it is a suggestive marker for NK cells, and CD16 binds the FC region of the IgG. Macrophages have CD14 receptor for PAMPs, pathogen-associated molecular patterns, like lipopolysaccharides. Then it has CD40, it has CCR5, MHC2 for CD4 helper cells, B7, FC, and C3b receptor for opsonization. Okay. And hematopoietic stem cells have CD34. Okay. Now, what is anergy? It is the state during which a cell cannot become activated by exposure to its antigen. So T and B cells become anergic when exposed to their antigen without the co-stimulatory signal, that is signal 2, that is the B7 of B cells binds to the CD28 of the CD20 8 of the T cells, right? Right, the B7 binds to the CD28 of the naive T cells. That is the secondary signal. So whenever there is no secondary signal, okay, so then the T and B cells are not going to stimulate, and it is another mechanism of self-tolerance. Because if it is a self-antigen, then also the T and B cells won't get activated. They become allergic, and it is seen in HIV. Right? As we all know, HIV is an immunodeficient state, right? So this is how it is immunodeficient. Okay. Now the passive versus active immunity. So passive immunity is receiving the preformed antibodies. We give preformed antibodies to the patient, but in active there is exposure to exogenous antigens, right? So the patient comes in contact with the antigens from the environment. Here we give preformed antibodies in the hospital setting in passive immunity. So obviously if we are giving the preformed antibodies, the onset of passive immunity is going to be rapid, and that of the active immunity is going to be slow. And duration of passive immunity is short span of antibodies. So it is the half-life of antibodies is 3 weeks. So for 3 weeks we are going to get immunity. Okay. And for active immunity it is long-lasting protection. So it has also memory, because the patient has received antigens from the environment, and body is going to activate T cell and then B cell, and then B cell is going to be transformed into plasma cell that is going to secrete immunoglobulins, and also memory cells that is going to help us with the memory of that antigen for future scenarios, right? So there's going to be long lasting protection. Examples of passive immunity are IgA in breast milk, in maternal IgG crossing the placenta, right? So the patient, that is the fetus, it's not a patient, but let's call him the fetus the patient. The patient is receiving IgA and IgG, right, from the mother. So that is preformed antibodies, right, and antitoxin, right, and humanized monoclonal antibody. Okay. And active immunity's examples are natural infection. The patient received antigens from the environment, or we give vaccine, that is inactivated antigen, right, and or toxoid, that is the protein component of the antigen that is inactivated through heat or through acid or something like that. Right. Now after exposure to, so examples of passive immunity are exposure to tetanus toxin, right, HBV, varicella, rabies virus, botulinum toxin, or diphtheria toxin. Unvaccinated patients are given preformed antibodies. Okay, so in all these cases, to heal very rapidly before dying, so tetanus toxin, HBV, varicella, rabies, botulinum toxin, and diphtheria, we have to give. The body can't make antibodies towards these infection in a quick time. So the pathogenesis of these antigens are a lot quicker than the speed at which our body can form antibodies towards it. So we have to give preformed antibodies to these patients. Okay, so yeah, this is the mnemonic which can help you remember. And in active immunity, we can combine passive and active immunization can be given for hepatitis B or rabies exposure. So in rabies, as we know, we give anti-rabies vaccine. Okay, so we give antibodies to it, antibodies for rabies, as well as we give inactivated rabies virus as an active immunity. Right? In active immunity, we are generating new immunity. Right? That's all you need to remember. in passive immunity, we are just transferring someone else's immunity to the required patient. Okay. Now the vaccination. So vaccination induces an active immune response. Okay. And that can be humoral or cellular to specific pathogens. Okay. So the first is live attenuated vaccine. So microorganisms rendered non-pathogenic. They are rendered non-pathogenic but retains the capacity for transient growth within inoculated host. So it can replicate within the host, but it cannot be pathogenic. Okay. It can okay. MMR and varicella vaccines can be given to people with HIV without evidence of immunity if CD4 plus cell count is more than 200 cells per millimeter cube. Okay. So if CD4 is more than 200, we can safely give the live vaccines MMR and varicella to the HIV patients. Okay. And the pros of live attenuated vaccines are induces cellular and humoral responses. It induces both the response, cellular as well as humoral. It induces strong, often lifelong immunity. In cons, it may revert to virulent form. And it is contraindicated in pregnancy and patients with immunodeficiency. Okay. And the mnemonic to remember live vaccines are: attention teachers, please vaccinate small beautiful young infants with MMR regularly. So that is attention for adenovirus, that is given to the military recruits. Okay. It is given to military recruits. That can be differentiating factor in the questions. The history of the question will give that it is or if it is asking about the adenoviral infection, it will state that it is amongst the military recruits, it is among the soldiers, and they were not vaccinated against that particular pathogen. So that is adenovirus. Okay. Attention teachers. Teachers is typhoid oral vaccine. Please vaccinate small beautiful young infants. So polio, that is Sabin vaccine, that is again oral vaccine. There is oral vaccine. Then varicella, chickenpox, smallpox, BCG, yellow fever, influenza, which is intranasal. Okay, yeah, it is here, intranasal, and with MMR regularly. So MMR and rotavirus. So all of those vaccines which are given through orally or through intranasally, they are all live vaccines. So and vaccines which are given intramuscularly and intravascularly, they are all killed vaccines. Right? So again, it is adenovirus, attention teachers, typhoid, please vaccinate small polio in oral, that is Sabin, vaccinate varicella, smallpox, beautiful BCG, young infants, yellow fever, and influenza with MMR regularly, rotavirus. Okay. Now the killed or inactivated vaccine. So pathogen is inactivated by heat or chemicals. Okay, pathogen is inactivated. Maintaining the epitope structure on surface antigens is important for immune response. Okay, but mainly induces a humoral response. So the pros are safer than live vaccines because they are killed vaccines. They are not going to revert to any virulent form. And cons is there is weaker cell-mediated immune response. Okay, and booster shots are usually required. So the vaccine has to maintain the epitope structure. Even if the pathogen is inactivated, it has to have a normal epitope structure in order to induce an immune response. Okay. And the mnemonic is a trip could kill you. So this is Hep A, typhoid, but this time it's not oral as it is a killed vaccine. It is going to be intramuscular type. So that is VI polysaccharide typhoid vaccine. Then a trip, rabies, again influenza but the intramuscular one, and again polio P, that is salt, that is the intra injectable, okay, intravascular. Okay, could kill you. So K is for the Salk type of the polio vaccine. Okay, so Hep A, typhoid intramuscular, then rabies, Haemophilus influenza intramuscular, and Salk polio. Now the subunit recombinant polysaccharide and conjugate. So all use specific antigens that best stimulate the immune response. So here the specific antigens are directly used to stimulate the immune response, but it can be its subunit of that pathogen, a recombinant DNA of the pathogen, or polysaccharide, or conjugate of all of them. So the pros are targets specific epitopes of antigens, lower chances of adverse reactions, okay, because it is targeting the specific epitopes. No other epitopes of antigens are targeted, so there is less chances of adverse reaction. And cons are expensive, weaker immune response. So examples are HPV, then acellular pertussis, and Neisseria. Neisseria meningitis, the various strains of Neisseria meningitis, and if you remember that sketchy image, the kid, it was of a dorm room where one of the kid was infected with Neisseria meningitis. So in that sketch it was shown that the other kids living in the dorm rooms are to be given the Neisseria vaccine and also rifampin as a prophylaxis, right? Then there's Streptococcus pneumonia. That is through PPSV23 polysaccharide, primarily T cell independent. If it is PPSV23, if it is PCV13 conjugated, it is T cell dependent. Okay, and the last is Haemophilus influenza type B and herpes zoster. All of them are from H, just except Neisseria meningitis and Streptococcus pneumonia. Okay, so HPV, okay, Neisseria meningitis, then strep pneumo, H influenza type B, and herpes zoster. So denatured bacterial toxin denatured bacterial toxin with intact receptor binding site. Okay, so that stimulates immune system to make antibodies without potential for causing any kind of disease. So there is everything intact, but just the bacterial toxin that is actually going to induce an immune response, that is denatured. So there is intact receptor 2 that is going to bind to its appropriate site and stimulate immune system to make antibodies. Pros are protect against the bacterial toxins. Cons are antitoxin levels decrease with time. Thus, booster shots may be needed, right? Whenever we have the patients come in with trauma or road traffic accident, we give booster shots of tetanus, right? Because the antitoxin levels decrease with time. So the examples are Clostridium tetani and Corynebacterium diphtheria. mRNA, a lipid nanoparticle, delivers mRNA, causing cells to synthesize foreign protein. Example, spike protein of SARS-CoV-2. It induces cellular and humoral immunity. Delivers mRNA. So the pros are high efficacy. It is safe in pregnancy. The COVID vaccine is safe in pregnancy. Cons are local and transient systemic. Fatigue, headache, and myalgia reactions are common. So we all took Covishield mostly in India, right? So as we know, there was fever and myalgia and headache for 1 or 2 days. So that is the systemic reactions, and rarely myocarditis and pericarditis, particularly in young males. So there is also cases of myocarditis and pericarditis associated with the COVID vaccines, but it is studied for now. It is not proven or anything like that. Now the hypersensitivity reactions, there are 4 types: A, B, C and D. A is for anaphylactic and atopic. B is antibody, or type 2 is antigen antibody. There are 2 things, antigen and antibody, so that is type 2. In type 3 there are that is delayed, so type 3 is immune complex mediated. So there are 3 things in the type 3: antigen, antibody and complex. Okay, so it is type 3. And type 4 is delayed, cell mediated type, which there is no antibody involvement, there is just T cells. So type 1, 2 and 3 are all antibody mediated. Type 1 hypersensitivity, anaphylactic and atopic. There are 2 phases. So immediate antigen immediate and late. Immediate involves antigen crosslinks, preformed IgE on pre-sensitized mast cell. Okay. Antigen crosslinks, preformed IgE on pre-sensitized mast cells, that will cause immediate degranulation, release of histamine, tryptase and leukotrienes. Histamine is a vasoactive amine, tryptase is a marker of mast cell activation, and leukotrienes. So the mast sorry for that. So there's always there is pre-sensitized mast cell. Okay, now the antigen crosslinks with the IgE found on the surface of the mast cells. So these are the IgE, allergen specific IgE, and allergen or antigen binds on the IgE. So that leads to immediate mast cell degranulation, and histamine, tryptase and leukotrienes are released. That is the immediate phase. In the late phase, there is chemokines, secretion of chemokines that attract inflammatory cells, example eosinophils, and other mediators from the mast cell that will lead to inflammation and tissue damage. Okay. So type 1 is first and fast. It is clinically associated with skin test, and skin test or blood test, that is ELISA for allergen specific IgE. We can test type 1 hypersensitivity either with skin test. If we perform a skin test with the suspected antigen, there is going to be immediate redness, swelling and increase in temperature at that local site. And we can also perform ELISA for allergen specific IgE. Okay. We cannot determine the allergen type, but we can find the IgE, specific IgE acting on that allergen through ELISA. ELISA is for finding out the antibodies, right, not the antigens. An example of type 1 hypersensitivity are anaphylaxis, food, drug or bee sting allergies, or allergic asthma. Type 2 hypersensitivity, the antibodies bind to cell surface antigens or extracellular matrix. So that will cause cellular destruction, inflammation and cellular dysfunction. Cellular destruction, inflammation and cellular dysfunction. So antibodies bind to cell surface antigens or extracellular matrix. So if this is the antigen, antigens have surface antigens, okay, or surface molecules, and the antibodies are bound to the NK cells, that are natural killer cells. So those antibodies are going to act directly on the surface antigens. So there are two things, antibody and antigen. So it is type 2 hypersensitivity, and it is antibody dependent cellular cytotoxicity. Okay, so one of the three things can happen. Either there is cell destruction, in which this cell is opsonized by antibodies or killed by antibodies, okay, and there can be inflammation, there can be binding of antibodies, and then it can activate the complement system. That is a long process. It can activate complement system, and then those complements will mediate the inflammation, or there can be cellular dysfunction. So the antibodies bind to the cell surface receptor, and there will be abnormal blockade of the process within the antigens. Okay. So let's see them in detail. So in cellular destruction, cell is opsonized by antibodies, okay, leading to either phagocytosis or activation of complement system or NK cell killing. So either phagocytosis or NK cell killing. So natural killer cells will directly kill the antigen. So that is antibody dependent cellular cytotoxicity. And phagocytosis can also lead to activation of complement system activation. So eventually complements are going to kill the antigen. And examples of these are autoimmune hemolytic anemia, immune thrombocytopenia, transfusion reactions, and hemolytic disease of the newborn. Basically all the diseases where the cells are decreasing in the number, right? In hemolytic anemia, the RBCs are decreasing, thrombocytopenia, platelets are decreasing, transfusion reactions, the recipient hemoglobin molecules are decreased, hemolytic disease, there is decrease in cell. Okay, so basically the cells decrease. Now in inflammation, whenever the antibody binds to the surface antigens, it is going to activate its own complement system and Fc receptor mediated inflammation. Okay, so there is going to be activation of complement system, which is going to mediate the inflammation. And the examples of it are Goodpasture syndrome, rheumatic fever, and hyperacute transplant rejection. Okay. And as there is activation of complement system, the complements are going to decrease in the body. Okay, there is reduction in the complements in serum. And in hyperacute transplant rejection, there is due to binding of HLA antibody. Okay. In cellular dysfunction, the antibodies bind to cell surface receptor, and abnormal blockade or activation of the downstream process. It can dysregulate the downstream process. It can either block the downstream process or it can activate the downstream process. So the examples are myasthenia gravis, Graves' disease, and pemphigus vulgaris. Right. So in myasthenia gravis, the autoantibodies are going to bind to the postsynaptic acetylcholine channels. So it is going to form dysfunction of those postsynaptic acetylcholine channels. Right. In Graves' disease and pemphigus vulgaris too, similarly. Okay. And do you find what are the tests for type 2 hypersensitivity? It is direct Coombs test detects antibody attached directly to the RBC surface, or indirect Coombs test where detects presence of unbound antibodies in the serum. Okay. Coombs test detects antibodies attached to the RBC, and indirect Coombs test detects presence of unbound antibodies in the serum. The type 3 hypersensitivity. So now type 3 hypersensitivity, it consists of immune complex, antigen and antibody. So there are three things. Mostly it is IgG antibodies. These complex activate the complements which attracts neutrophils. Neutrophils releases lysosomal enzymes, and it can be associated with vasculitis and systemic manifestations. So these antigen antibody and complement complexes, they attract neutrophils, and neutrophils, as you know, are going to release these enzymes, lysosomal enzymes, which is going to digest the pathogen. Okay. So now there are two examples of type 3 hypersensitivity. The first is serum sickness, the prototypic immune complex disease. So antibodies to foreign proteins are produced, and 1 to 2 weeks later antibody antigen complex is formed and deposit in tissues. That is going to form complement activation and inflammation and tissue damages. So as there is complement activation, there is going to be decreased serum C3 and C4. Right. So antigen antibody complex is formed there and then itself, but after 1 to 2 weeks there is antigen antibody complex binding with the complements, right? There is complement activation after 1 to 2 weeks. Okay. And second is Arthur's reaction, a local subacute immune complex mediated hypersensitivity reaction. So intradermal injection of antigen into pre-sensitized individual leads to immune complex formation in the skin at the local site. Enhanced local reaction to a booster vaccination, characterized by edema, fibrinoid necrosis and activation of complement, and there is going to be localized inflammation at the site of injection. Okay, there is going to be redness, there is going to be increased heat, swelling and all that. In type 3 reaction, imagine an immune complex has three things stuck together: antigen, antibody and complement. Examples are all the diseases with complement complexes. Okay. SLE, rheumatoid arthritis, reactive arthritis, polyarthritis nodosa, post-streptococcal glomerulonephritis and IgA vasculitis. So as you know, rheumatoid arthritis and post-streptococcal glomerulonephritis, they all are caused some time after the pathogen attacks the body, right? So that is the time taken for the complement activation. In the fever, urticaria, arthralgia, proteinuria, lymphadenopathy occurs 1 to 2 weeks after antigen exposure. So these are the symptoms of the type 3 hypersensitivity: fever, urticaria, arthralgia, proteinuria like in PSGN, and lymphadenopathy. Okay. It occurs 1 to 2 weeks after antigen exposure. Serum sickness-like reactions are associated with some drugs, may act as haptens. Serum sickness-like reactions are associated with some drugs, may act as haptens, penicillin, monoclonal antibodies, and infections, for example, hepatitis B. So with these haptens, that is penicillin and monoclonal antibodies, and infections like Hep B, there is going to be serum sickness-like reaction. Not exactly the serum sickness, but serum sickness-like reactions. So that is very less severe than serum sickness. Less severe than the actual serum sickness. Okay, there is only fever, rash and urticaria and purpura, but they are very mild. And if you want to remember these examples, just remember the diseases caused by the autoantibodies. Autoantibodies or cross antigen reactivity. like rheumatoid arthritis, PSG, and they both have cross antigen reactivity. Reactive arthritis has cross antigen reactivity. SLE has, as you know, there is complement activation and complement accumulation. There is antibodies towards the nuclear proteins, right? Now the type 4 hypersensitivity. So the type 4 hypersensitivity, there are two mechanisms in type 4 hypersensitivity, each involving the T cells. So the first is direct cell cytotoxicity. CD8 plus cytotoxic T cells kill the targeted cells directly. That's it. And second is inflammatory reaction. So CD4 plus T cells recognize the antigen and release inflammation inducing the cytokines. Okay, so this is the antigen presenting cell presenting the antigen to the Th1 cell, and that Th1 cell is going to release cytokines that is going to activate the macrophage and it is going to engulf the antigen. Responses do not involve antibodies. Examples: contact dermatitis, that is poison ivy, and nickel allergy, and graft versus host disease. Tests: PPD for TB infection, patch test for contact dermatitis, and candida skin test for T cell immune function. So there are 4 T's you have to remember, that is T cell, transplant rejections, TB skin test, and touching, that is contact dermatitis. And the examples of type 4 hypersensitivity, that is graft versus host disease, examples of GVH disease. In kidneys it is glomerular sclerosis, in liver it is vanishing bile duct, and in heart it is accelerated atherosclerosis. Okay, coronary CAD is coronary artery disease. So in heart it causes accelerated coronary artery disease, right? And you also have to remember these clinical associations, that is candida skin test. It is the mechanism of the candida skin test, tuberculin test, and patch test for allergy. Now the immunologic blood transfusion reactions. So first is allergic anaphylactic reaction that occurs immediately with transfusion. It is the pathogenesis includes type 1 hypersensitivity, reaction against plasma proteins in transfused blood. And IgA deficient individuals should receive blood products without IgA. Now let's see why is it so, that the donor blood has these immunoglobulins including IgA, right, and donor plasma proteins. So the IgA are going into the host blood. So there are in host blood there is IgE that can attack IgA if the individual is already IgA deficient, right? So if the individual is IgA deficient, it is going to treat the foreign IgA as an antigen, and it is going to attack those foreign IgA, right? So that can lead to anaphylactic reaction. So IgA deficient individuals should receive blood product without any IgA. Okay. And timing is within minutes to 2 to 3 hours due to release of preformed inflammatory mediators and degranulating mast cells. So IgA, IgE are present on the mast cells. This is the mast cells, and as soon as IgE is bound to any antigen, there is degranulation of mast cell, which is going to release histamine, tryptase, and leukotrienes, which will mediate the anaphylactic reaction. Clinical presentations are allergies, that is urticaria and pruritus, anaphylaxis, that is wheezing, hypotension, and respiratory arrest and shock too. Okay. Allergies and anaphylaxis. Second is acute hemolytic transfusion reaction. It is due to type 2 hypersensitivity reaction. Typically causes intravascular hemolysis. So there is going to be decrease in haptoglobulin. And ABO, blood group incompatibility, as we discussed here. In type 2, due to the cellular destruction phenomenon, there is going to be decrease in the cells. And this is a transfusion reaction, decrease in RBC. Okay. During transfusion or within the 24 hours due to preformed antibodies. The clinical presentation is fever, hypotension, tachypnea, tachycardia, flank pain, hemoglobinuria if it is intravascular, jaundice if it is extravascular hemolysis. So the donor blood has A and B group antigens, and host blood can have host anti-A, anti-B, IgG or IgM. Febrile non-hemolytic transfusion reaction, cytokines created by donor WBC accumulate during storage of blood products, right? And then when that stored blood product is given to a recipient, those cytokines created by the donor WBC is going to attack the recipient's blood. Okay. Reactions prevented by leukoreduction of blood products if it is to be stored for a longer time. It is a febrile non-hemolytic. Okay. So those cytokines are just going to mediate fever and acute inflammation, right? So cytokines that are IL-1, IL-6 and TNF-alpha, they are just going to mediate inflammation. So the symptoms of inflammations are going to be seen, that is fever, headaches, chills and flushing, more common in children. So within 1 to 6 hours due to preformed cytokines. Allergic reaction was due to preformed Allergic reaction was due to preformed inflammatory mediators. Acute hemolytic reaction was due to preformed antibodies. And febrile non-hemolytic transfusion reaction is due to preformed cytokines. Okay. So donor WBC releases preformed cytokines. Transfusion related acute lung injury. So there is two-hit mechanism. So initially in patients of respiratory disease or any lung pathology, there are neutrophils that are already sequestered in, primed in pulmonary vasculature due to recipient risk factors. Okay. So now those neutrophils are activated by a product, that is anti-leukocyte antibodies, in the transfused blood and release inflammatory mediators. Right. So increased capillary permeability and can cause pulmonary edema. Right. So donors, here the role of donor is to provide anti-leukocyte antibody. So everything else is just due to the risk factors of the recipient, because as the recipient is having risk factors, there is also going to be neutrophils accumulated in the pulmonary vasculature. So when this anti-leukocyte antibody from the donor cells reaches the host neutrophils, it is going to cause inflammatory reaction, right? And that inflammatory reaction, as the signs of inflammation we see, we can see that it is edema and swelling and all that. So that due to edema, it is going to cause pulmonary edema. So and increased capillary permeability. It occurs in within minutes to 6 hours. Only acute hemolytic transfusion reaction can occur within 24 hours. All other anaphylactic, febrile, non-hemolytic and trally, all three of them occur within 2 to 3 or 6 hours. Okay. And the examples are respiratory distress, non-cardiogenic, pulmonary edema. It presents as respiratory distress, non-cardiogenic, pulmonary edema. So there are normal heart signs, but there is still pulmonary edema. The last is delayed hemolytic transfusion reaction. So amnestic response to a foreign antigen on donor RBC. response to a foreign antigen on donor RBC, that is RHD or other minor blood group previously encountered by recipient. Typically causes extravascular hemolysis, so there will be jaundice. Onset over 24 hours, usually presents within 1 to 2 weeks due to slow destruction by reticuloendothelial system. So reticuloendothelial system takes time to destroy these foreign antigens found on the donor RBCs. So those foreign antigens on donor RBCs can be any minor blood group, or even the Rh factor can act as a foreign antigen. And the recipient should have previously encountered that foreign antigens. Okay, so that this delayed hemolytic transfusion reaction occurs when the foreign antigens comes in contact for the second time. Okay, and they are generally self-limited, clinically silent. There can be mild fever or hyperbilirubinemia because it is extravascular hemolysis. These are the autoantibodies associated with different disease. You just have to remember, and you will remember it with continuous use of these autoantibodies. It's not a big deal. So myasthenia gravis, anti-postsynaptic acetylcholine receptors, and if it is presynaptic and voltage gated calcium channel, it is going to be Lambert-Eaton myasthenic syndrome. Beta-2 microglobulin glycoprotein, APLA, ANA, that is non-specific and mostly associated with SLE. If ANA is positive, then we refer the case to the rheumatological department, because as soon as the ANA comes positive, it can be number of things, like it can be any SLE or mixed CTD or Sjogren's syndrome, scleroderma, anything like that. So this is the gateway to the rheumatological department. Then this anticardiolipin and lupus anticoagulant, that is in SLE and APLA. Anti-dsDNA, anti-Smith, again SLE. Antihistone is in drug-induced lupus. Anti-U1 RNP, ribonucleoprotein, it is in mixed CTD. Rheumatoid factor, IgM, antibody against IgG Fc region, and anti-CCP, more specific, are for rheumatoid arthritis. So IgM antibody attacks the Fc region of the IgG. Anti-Rho and anti-La, that is SSA and SSB for Sjogren's syndrome. Anti-SCLER70, that is for scleroderma, obviously, against DNA topoisomerase. Okay, DNA topoisomerase breaks the DNA strands, right? If it is 1, it breaks single-stranded DNA. If it is DNA topoisomerase 2, it is going to break double-stranded DNA. Anti-centromere for limited scleroderma. It is part of the CREST syndrome. Anti-synthetase, that is anti-Jo1 or anti-SRP or anti-helicase. It is for polymyositis or dermatomyositis. This one is important, anti-Jo1. And anti-mitochondrial for primary biliary cholangitis. Okay. That is AMA antibody, as we saw in gastroenterology system, like AMA is positive in PBC, right? And in PSC, primary sclerosing cholangitis, there was no AMA positivity, but it was just there was ANCA positivity, and it was associated with ulcerative colitis in males. Right. Now the anti-smooth muscle or anti-liver kidney microsomal 1, anti-LKM1 for autoimmune hepatitis. Myeloperoxidase anti-neutrophil cytoplasmic antibody, that is MPO-ANCA or PNCA. They are associated with microscopic polyangiitis, eosinophilic granulomatosis with polyangiitis, ulcerative colitis, and primary sclerosing cholangitis. I'm pretty sure this is, I think, vaginous. I may be incorrect. And this is, I think, Sjögren's. I don't know. The ulcerative colitis and PSC are important. And the C ANCA, that is cytoplasmic ANCA with granulomatosis with polyangiitis. Okay. Then there's anti-phospholipase A2 receptor with membranous nephropathy, anti-hemidesmosome with bullous pemphigoid. So if there's antibody towards hemidesmosome, it is going to separate the two layers of the skin, and there is going to be liquid or secretion between those two layers, causing bullous pemphigoid. This will cause a bulla. Okay. An antidesmoglein, antidesmosome, that will cause pemphigus vulgaris. Hemidesmosome, bullous pemphigoid, and antidesmosome, pemphigus vulgaris. Antithyroglobulin and antithyroid peroxidase in Hashimoto's thyroiditis. So antithyroid peroxidase, so T3 is not going to convert into T4. So T3's levels are going to rise. That will cause hyperthyroidism. So, and anti-TSH receptor, that is going to cause Graves' disease, hyperthyroidism. And IgA, anti-endomycyl IgA, anti-TTG, IgA and IgG deaminated gliadin peptide are all seen in celiac disease, as we saw in GIT. Anti-glutamic acid decarboxylase, islet cells cytoplasmic antibodies are seen in type 1 diabetes mellitus. Okay, anti-glutamic acid decarboxylase and islet cells cytoplasmic antibodies. So antibodies act on the islet cell, causing its destruction. Antiparietal cell and anti-intrinsic factor is pernicious anemia. And anti-glomerular basement membrane, it's Goodpasture syndrome. Okay. Now the immunodeficiencies, that are B cell, T cell, and both B and T cell. Right. B cells are B cells include Bruton's A gamma globulinemia, selective IgA deficiency, and common variable immunodeficiency. Right. So defect in Bruton tyrosine kinase gene, that is a BTK, a tyrosine kinase gene. So there will be no B cell maturation, right? And as there is no B cell maturation, there is not going to be enough B cells, right? And it is X-linked recessive, and that means it is higher in boys. Okay. Its presentation of Bruton A gamma globulinemia is recurrent bacterial and enteroviral infections, right? Bacterial and enteroviral infections after 6 months. Because for the first 6 months there is maternal IgG from the placenta. So only IgG can cross the placenta, right? We know that. And the findings are absent B cells in peripheral blood, decreased immunoglobulins of all classes, because as there is no B cells, there is no class switching going on of the immunoglobulins in the B cells, so there will be decreased immunoglobulins of all classes. And as the B cells are absent, there will be absent or scanty lymph nodes and tonsils, because those are the sites where B cells mature, right? So if there are no B cells, we don't need those sites. Primary follicles and germinal centers are absent. Okay. And live vaccines are contraindicated, because live vaccines can induce an virulent infection in them. Yeah. And giardiasis is very common too. With Bruton's A gamma globulinemia, and this is the point to remember, that all immunoglobulins are decreased. Okay. And selective IgA deficiency, the name itself says this, there is just deficiency of IgA with normal IgG and IgM levels. So the cause is unknown, and it is the most common primary immunodeficiency. Right? It is not due to any acquired cause or any congenital cause, but it is primary immunodeficiency. You have to remember all the A's, like it's a majority is asymptomatic, can see airway and GI infections. It is autoimmune. It can have atopy. It can have anaphylaxis to IgA in blood products. Right? We discussed this, that if an IgA deficient individual is getting a blood which will have IgA antibodies, so those foreign IgA antibodies will be treated as a foreign antigen, right? So the recipient's IgE are going to act on the donor's IgA and produce anaphylactic reaction. Right? And there's increased susceptibility to giardiasis. Wherever there is IgA deficiency, there's susceptibility to giardiasis, and it can also cause false negative celiac disease. So celiac disease is associated with IgA deficiency, but it will be falsely negative. Now the third is common variable immunodeficiency. There's defect in B cell differentiation. Right? Here is similar to Bruton's A gamma globulinemia, where there was no B cell maturation. Here there is defect in B cell differentiation. It can cause causes unknown in most cases. Okay, it may present in childhood, but usually diagnosed after puberty. Okay, and there's increased risk of autoimmune disease, bronchiectasis, lymphoma, and sinopulmonary infections. Right? Autoimmune bronchiectasis, lymphoma, and sinopulmonary infections. Findings are decreased plasma cells because of decreased B cell differentiation. So the B cell is not able to differentiate into plasma cell and mammary cell. Right? And the immunoglobulins are going to decrease as well because there is lack of matured B cell. Right? If plasma cells are decreased, obviously immunoglobulins are decreased as well. Right, so these three disorders are for B cell. Okay, and if whenever there is B cell deficiency or B cell defect, mostly the infections are going to be of bacterial type, right? And if it's a T cell deficiency, there will be viral and fungi and parasitic infections more predominant. But for B cell, you can remember these bacterial and enteroviral infections for Bruton's, then airway and GI infections, again airway and GI infections for IgA deficiency, and there's sinopulmonary infections and lymphomas. Lymphomas arise from the B cells. So the undifferentiated B cells accumulate and form a lymphoma. Lymphoma, sinopulmonary infections, both are due to B cells, right? Now the T cell disorders. So in T cell disorders, we have 4 disorders that are thymic aplasia, there is Digeorge syndrome, then IL-12 receptor deficiency, and autosomal dominant hyper IgE syndrome, that is Job syndrome, and chronic mucocutaneous candidiasis. So thymic aplasia, there's 22q11 microdeletion. There's failure to develop third and fourth pharyngeal pouches. So there's absent thymus and parathyroid glands. Right, third and fourth pharyngeal pouches are destined to form thymus and parathyroids, but they are absent. So Digeorge syndrome is thymic, parathyroid, and cardiac defects. Right? Thymic, parathyroid, and cardiac defects. And velocardial facial syndrome is palate, facial, and cardial defects. Okay, palate, facial, and cardio defects. So you can figure it out from the name itself. Facial, that is facial; cardio, that is cardio defect; and velo is palate. Okay. Now the what's the mnemonic for remembering the thymic aplasia? So that is catch-22 syndrome. C for the cardiac defects, A for abnormal facies, T is for thymic hypoplasia, C is again for cleft palate, and H is for hypocalcemia. So cardiac defects are mostly conotruncal abnormalities, that are those three: the tetralogy of Fallot, truncus arteriosus, and there was another T there, but transposition of great arteries, I think. Okay, those are the three conotruncal abnormalities. Then the A is for abnormal facies, T is for thymic hypoplasia. So now if the thymus is hypoplastic, there is no place for T cells to mature, right? So there is going to be a T cell deficiency, matured T cell deficiency. And as I said, that the B cells are mostly associated with the bacterial infections, whereas the T cells are mostly associated with the viral, parasitic, and fungi infections. So if T cells are deficient, there is going to be recurrent viral and fungal infections. Right. And there's cleft palate, there's hypocalcemia secondary to parathyroid aplasia, right? Because PTH can't be secreted and it can't regulate the calcium in the serum, right? So the hypocalcemia is signified by tetany. Oh, that is okay. Findings are decreased T cells, right? Decreased parathormone and decreased calcium. And thymic shadow is absent on chest X-ray. Mostly an X-ray will be given where there is no thymus shadow, and the age of the patient would be given less than 2 years or less than 6 years or anything like that, right? Because that's when you should be able to see the thymic shadow, right? IL-12 receptor deficiency. So IL-12 is secreted from the macrophage, right? And the function of IL-12 was to differentiate TH into Th1. Yeah. And it also activates NK cells, natural killer cells. And so you just have to, all the down process are inhibited if the IL-12 receptor deficiency is there. So TH will not be differentiated into Th1, and Th1 will not further secrete interferon gamma. Okay. And interferon gamma was responsible for granuloma formation in mostly tuberculosis, right? Because it was stimulating macrophage to stimulate to form the granuloma, right? So if interferon gamma is not released, there will not be any type of granuloma formation. There will not be any kind of immune response towards the tuberculosis, right? So Th1 response is decreased. It's autosomal recessive. Okay, so Th1 response is decreased. Disseminated mycobacterial and fungal infections, and it may present after administration of BCG vaccine, because BCG vaccine we are giving inactivated tuberculosis pathogens, right? So if we don't have an immune response towards that tuberculosis pathogen we gave in the vaccine, there's going to be adverse reaction to that, right? There is not going to be any kind of immune response because we can't form granuloma against it. Okay. And there's disseminated mycobacterial and fungal infections. Obviously, because it is a T cell deficiency, there's going to be mycobacterial and fungal infections. Mycobacterial, that is TB. And interferon gamma findings are interferon gamma are decreased. Most common cause of Mendelian susceptibility to mycobacterial disease. Now, the autosomal dominant hyper IgE syndrome. So deficiency of Th17 cells due to STAT3 mutation. Okay, there's deficiency of Th17 due to STAT3 mutation. So there's impaired recruitment of neutrophils to the site of infection. Now let's see those T cell differentiation chart. Right, here's the Th17. It induces neutrophilic inflammation, right? It recruits neutrophils via IL-17, IL-21, and IL-22. Right? So there's impaired recruitment of neutrophils to the site of infection. So the presentation of Job syndrome is by cold, non-inflamed because we can't recruit neutrophils, right? The staphylococcal abscesses, retained baby teeth, coarse faces, dermatological problems like eczema, and it is because of increased IgE, and there is bone fractures from minor trauma. Okay, staphylococcal abscesses develop due to impaired recruitment of neutrophils, right? There is not enough fighting response, so that leads to abscess formation. Abscess formation mostly takes place in the patient with immunosuppression or weakened immunity, right, or old age, any of them. So the findings are increased IgE, increased eosinophils, and learn the ABCDEF to get the job. A is for abscess, B for baby teeth, C for coarse faces, D for dermatological problems, that is eczema, E for increased IgE, and F is for the fractures from minor trauma. Right. And chronic mucocutaneous candidiasis, that is T cell dysfunction, impaired cell-mediated immunity against Candida species. Specifically, Candida species is a classic form caused by defects in AIRE. So AIRE was responsible for the negative selection in Thymus, right? Yeah, in Thymus, yeah. During the T cell differentiation, we learned about the positive and negative selection. So AIRE gene transcription was the role of AIRE was to present endothelial epithelial dendritic cells to the T cells, and those T cells that reacted to those epithelial cells were undergoing apoptosis to prevent autoimmunity in future. So that was the role of the transcription factor AIRE, right? So if there's a defect in AIRE, the T lymphocytes were able to escape. Right. Self-reacting T cells were able to escape. As T cell dysfunction impaired cell-mediated immunity against Candida species. Persistent non-invasive Candida albicans infections of skin and mucous membranes. Right? It's non-invasive Candida infection of skin and mucous membranes. The findings are absent in vitro T cell proliferation in response to the Candida antigens. If we perform Candida skin test, it's going to be negative, and absent cutaneous reaction to Candida antigens. Right. And it's supposedly due to IL-17 and IL-17 receptor defects. Now the most important one, SCID. So SCID has several types, including defective IL-2 receptor gamma chain, most common X-linked recessive. Then there's adenosine deaminase deficiency, ADA deficiency, that is autosomal recessive, and RAG mutations, that is light chain recombination defect. Right? So if we are not able to recombinate the light chains, there is not going to be antigen specificity, right? So yeah, there are three types. First is IL-2 receptor defective, ADA deficiency. The third is RAG mutations. Okay. We learned about the RAG mutations in the central tolerance in the bone marrow, right? And presentations are a failure to thrive, and there's chronic diarrhea and thrush, and there's recurrent viral, bacterial, fungal, and protozoal infections. There's viral as well as bacterial infections because it is combined B and T cell disorders, right? and both cell-mediated and humoral immunity are decreased. Decreased T cell receptor excision circles, that are tracks. This is now a part of newborn screening for SCID, and to determine the T cell receptor excision circles, there's absence of thymic shadow, which represents T cell deficiency. There's germinal center absence, which represents B cell deficiency. Okay, and decreased T cells on flow cytometry. Right. So in B cell disorder, there was just absent or scanty lymph nodes and tonsils, because they are the site of the B cell maturation. Whereas in T cell disorder, there was thymic shadow absence. Right, but in B and T cell disorders, both will be absent. Right. And there's cytokine receptor defects. These are the cytokine receptor defects. And also a drug called cladribine, that is adenosine deaminase inhibitor, is going to present with similar symptoms as SCID. Okay. And also there's one more finding, that is increased deoxyadenosine concentration in urine and serum. Because if the adenosine is not deaminated, it is going to form deoxyadenosine. The second is ataxia telangiectasia. So there is defect in ATM gene, which will cause failure to detect the DNA damage. So DNA damage is occurring at all times in a normal individual, right? But we have a mechanism to detect the DNA damage and to chop off that damaged DNA out of our normal DNA. But due to defect in the ATM gene, there's failure to detect that DNA damage, and that will cause failure to halt the progression of the cell cycle. So cell cycle will go on with that damaged DNA, and that will cause accumulation of the mutations, and that is autosomal recessive. So the triad of ataxia telangiectasia are: first is ataxia, that is cerebellar defects, then there's spider angioma, then there's IgA deficiency. The three A's, that is ataxia, angioma, and IgA deficiency. And there's increased sensitivity to radiations. Okay, so the radiations will cause more telangiectasias to form. So the findings are increased alpha fetoprotein, decreased IgA, IgG, and IgE. So the age decreases, age decreases, lymphopenia, and cerebellar atrophy. This can be sometimes compared to the Friedreich's ataxia, where you can't figure out any atrophy in the cerebellum area on MRI, right? But in ataxia telangiectasia, it will have a similar presentation as Friedreich's ataxia, but in ataxia telangiectasia, you can figure out cerebellar atrophy on MRI. Right? There's increased risk of lymphoma and leukemia. Okay, so all the A's that you have to remember, that is ATM, there's ataxia, spider angioma, decreased IgA, increased alpha fetoprotein. Right. These are the A's, and then there are other things like lymphopenia, cerebellar atrophy, and there's lymphopenia, there's lymphoma, and leukemia. Now this, 1 second. Now, hyper IgM syndrome, it is the most commonly due to defective CD40 ligand on Th cells. So there is class switching defect because CD40 from B cell has to bind CD40 ligand on the Th cells in order to do the class switching, so that IgM can be initial IgM can be converted into different immunoglobulins, right? So there is class switching defect. It is X-linked recessive, and as there is class switching defect, there is going to be increased IgM, which is not able to convert into different immunoglobulins, so that would lead into deficiency of all other immunoglobulins. So the presentations are severe pyogenic infections early in life, opportunistic infections with pneumocystis and cryptosporidium, and CMV. There is upper respiratory tract infection, right? Lower respiratory tract, that will cause pneumonia through pneumocystis jirovecii, and there's cryptosporidium, so that is GI infection, and all pyogenic infections. There's normal or increased IgM, so the name itself suggests that is hyper IgM syndrome. There's decreased age, so that is IgE are decreased, just like Edexa telangiectasia, but it is here there is severe deficiency of IgE immunoglobulins, and there's failure to make germinal centers because it is B cell disorder too, just like it is a T cell disorder too. So the last is Wiskott-Ulrich syndrome. There's mutation in the WAS gene, WAS gene. So leukocytes and platelets unable to recognize actin cytoskeleton. There is a defective antigen presentation, and there is X-linked recessive. Not recognized, sorry, my bad. That's reorganized actin, right? Leukocytes and platelets are unable to reorganize the actin cytoskeleton within itself, so there is defective antigen presentation. And you just have to memorize these this mnemonic, that's water, Wiskott-Ulrich, there's thrombocytopenia, eczema, and recurrent infections, that is pyogenic recurrent infections. And there's increased risk of autoimmune diseases and malignancy. There's reduced to normal IgG and IgM. There is increased IgE and IgA. There's fewer and smaller platelets. So in SCID, there is all three kinds of infections: viral, bacterial, fungal, and even protozoal infections. In Edexa telangiectasia, you can easily differentiate it with presence of ataxia or telangiectasia, or those two are the very specific points for Edexa telangiectasia. And in hyper IgM syndrome, you can you can differentiate it with pneumocystis or cryptosporidium history. So cryptosporidium history would be like chronic diarrhea, right? And pneumocystis will be recurrent pneumonia infections, and that would be early in life. Okay. And there can be also symptoms of CMV infections like ring-like calcifications in the brain, and developmental delay in the brain, all that. And also you can differentiate Wiskott-Ulrich with thrombocytopenia, that is very significant here. And there's also eczema, right? Thrombocytopenia and eczema. And only in this condition the IgE and IgA are increased, right? The phagocytic dysfunction. First is leukocyte adhesion deficiency, lab type 1. So there is defect in LFA1 integrin protein, that is CD18, on phagocytes. So there is impaired migration and chemotaxis. Right. So integrin proteins bind to the ICAM1 on endothelium, right? So if integrin protein is defective, it is not able to bind to the ICAM receptors on an epithelium, and so therefore it cannot stick to the endothelium, and there is impaired migration and chemotaxis. Like the neutrophils can't reach the phagocytes can't reach the desired site of inflammation, right? And There's also a simple mnemonic of LAD. There's late separation of umbilical cord for more than 30 days. There's absent pus, and there's dysfunctional neutrophils. Absent pus because neutrophils and phagocytes are not able to reach the desired site, right? Because so that they are not able to generate an immune response as required, and so there will be absent pus, right? And there's dysfunctional neutrophils, which will cause recurrent skin and mucosal bacterial infections. Right. The late separation of umbilicus is the differentiating feature here. And there will be increased neutrophils in blood. See, this is the vessel. Here are the ICAM1 receptors. There are neutrophils here. In case of inflammation, they will try to stick it to the ICAM1s and reach the site of inflammation. This is the site of inflammation. But now neutrophils are increasing in response. Neutrophils are increasing in blood in response to the inflammation, but they are not able to bind to the ICAM1 receptors, right? Because of LFA1 integrin protein defect, right? So there will be increased neutrophil in the blood. It's just that they are not able to reach the site of infection. Okay. So there's absence of neutrophils at the infection sites, right? Here are neutrophils defined down. They are not able to reach that site. And there's impaired wound healing as well. Now the Chediak-Higashi syndrome. There's defect in lysosomal trafficking regulator gene. So this gene, list gene, is responsible for lysosomal trafficking. So it is responsible for transporting of different things around the cell. Like there are channels in the cell through which the proteins or the other substances travel, and that is regulated by this lysosomal trafficking regulator gene, right? And so there is microtubule dysfunction in phagosome lysosome fusion, right? There is microtubule dysfunction. If there is microtubule dysfunction, the substances are not able to reach their destination, right? They are steady at that point. They are producing the trafficking at that point of place. So the symptom presentation is plain, that is progressive neurodegeneration. All of these presentations are due to the things not reaching their destined place, right? If there's progressive neurodegeneration, there is lymphohistiocytosis. So the lymphocytes and histiocytes of the skin are not able to reach at their perfect place. They are being accumulated in the blood, right? There's albinism. It is supposedly that the melanocytes are not able to reach the skin, so that is why there's albinism. There's recurrent pyogenic infections. There's peripheral neuropathy because of the, I don't know, some kind of signals not reaching the appropriate nerves it was supposed to, so that might be causing some peripheral neuropathy or anything. It's the same reason neurodegeneration may be occurring. In histology, there's giant granules in granulocytes and platelets, right? So these are the giant cells. Right? There's pancytopenia as well. All kinds of cells decrease, right? And there's mild coagulation defects as well, because the coagulation factors might not be able to reach the sites. The last is chronic granulomatous disease. So there is defect in NADPH oxidase. So that leads to decreased reactive oxygen species, for example the superoxides, and there's decreased respiratory burst in neutrophils, and it is X-linked form most common. So these the most important function of these reactive species was to fight catalase positive organisms. Now, if the reactive oxygen species are deficient, or there is decreased respiratory burst in neutrophils, there is going to be increased susceptibility to catalase positive organisms, right? And there would be recurrent infections and granulomas. So which were the catalase positive organisms? Let's again revise those. It was Staph aureus, E. coli, Pseudomonas, Serratia, Candida, and many more, Aspergillus. We will discuss them again here. And there would be abnormal dihydrorhodamine test, that is the flow cytometry test, and there is decreased green fluorescence, so that is abnormal. It should have increased green fluorescence. And the nitroblue tetrazolium test, reduction test, fails to turn blue, right? So nitroblue tetrazolium test is failed, so that is a negative test. It does not turn it into blue. So we can say that NBT test is negative. Right, now if you guys remember, there was we talked about MPO deficiency, myeloperoxidase MPO deficiency. It is required by the peroxidase enzyme and all that. If you want to go back, here it is there is myeloperoxidase, contains a blue-green heme containing pigment that gives sputum its color, right? And it is required in this process of respiratory burst. Right. I don't know where exactly it is required, but it's required somewhere distal to the site where NADPH oxidase is required, right? Somewhere over here MPO might be required. Okay. Yeah, here it is. It was front of my eyes all the time. Here this is myeloperoxidase is required, sorry. Right. So in MPO deficiency, the NBT test was normal. Yet it was able to turn it into blue. Right? That is how you differentiate chronic granulomatous disease and myeloperoxidase deficiency. And there will be recurrent infections, there will be granulomas, because it is granulomatous disease, right? There is going to be lymphadenopathy, there is going to be pustules. Now the infections and immunodeficiency. So the first is bacteria. So if there's T cell deficiency, it is going to cause just sepsis. But if there's B cell deficiency, it can cause number of infections, right, which are encapsulated. So please shine my skis, that is Pseudomonas aeruginosa, strep pneumo, H. influenza, Neisseria meningitis, E. coli, then Salmonella, Klebsiella, and group B, Streptococcus. And if there's granulocyte deficiency, like in phagocytic dysfunction, right? The mnemonic is some bacteria produce no serious granules. So Staphylococcus, Burkholderia, Pseudomonas aeruginosa, Nocardia, and Serratia. And if there's complement deficiency, there's encapsulated species with early complement, right? We saw that in the complement section, that if there's early complement deficiency, there's mostly airway infections and all that, right? Like if there's early complement deficiency, there's recurrent pyogenic sinus, respiratory tract infections and all that, because C3b is used in clearance of antigen antibody complexes, and C3b is used in opsonization of encapsulated organisms, mostly the strep pneumo, which causes pneumonia, right? And if there's a terminal complement deficiency, there's mostly Neisseria bacteremia, okay, Neisseria meningitis, okay, and Neisseria with late complement, that is C5 to C9, that forms membrane attack complexes. Which are the viruses infecting the immunodeficient people? If there's T cell deficiency, there is CMV, EBV, JC, varicella zoster, chronic infection with respiratory and GI viruses. And there's not much virus infection with deficiency of B cells. Okay, B cells are mostly in bacteria, right? In viruses section, there's just decreased T cells. Okay, enteroviral encephalitis, polio, and poliovirus. Okay, and so the live vaccine is contraindicated in B cell deficiency. Nothing in granulocytes and complements. In fungi and parasites, again, T cell deficiency is more predominant. So it was going to cause Candida local infection, local mucosal infection, then PCP and Cryptococcus. Right? And in B cell deficiency, there's always giardiasis with deficiency of IgA, okay, that we saw here. Right. Giardiasis, then again here giardiasis is because of the deficiency of IgA, and giardia is a parasite. And with granulocyte deficiency, there is Candida, systemic infection of Candida. With if T cell deficiency, there was local Candida infection, but with decreased granulocyte, there is systemic Candida infection, also Aspergillus and mucormycosis. So if you can see here, there is Candida species, there is Candida albicans infections of skin and mucous membrane, so that is localized infection of Candida with T cell deficiency. And with phagocytic dysfunction, as you can see here, in chronic granulomatous disease, there is systemic Candida involvement. B cell deficiencies tend to produce recurrent bacterial infections, whereas T cell deficiency produce more fungal and viral infections. Okay, and also now let's discuss Retter's syndrome, that is reactive arthritis. So the only thing you have to remember is can't see, can't pee, and can't climb a tree. Okay, so can't see is conjunctivitis and uveitis. Okay, and can't pee is urethritis or UTI. And can't climb a tree, that is arthritis. Okay, or enthesitis or dactylitis. Or another term known as keratoderma blenoragicum. Okay, that is the hyperkeratinization of palms and soles. And what exactly is the pathogenesis? It is a post-bacterial infectious autoimmune condition. So antibodies attacking the bacterial infection also attacks all these things, that produces a lot of symptoms. So post-infectious AI condition after bacterial infection of GI tract or urinary tract. And it is a type 3 hypersensitivity. So there is antigen, antibody and complex formation. So there is going to be decreased C3, C4. Also, there's another disease I want you to know, that is severe congenital neutropenia. So there is congenital neutropenia that is very severe. So it causes recurrent infections, recurrent otitis media, then respiratory infections and skin infections, skin abscesses. And neutropenia is so severe that it is less than 200. And there's monocytosis. Now the last topic of the immune responses, that is transplant rejection. It is the most important topic of immune responses. Okay. And you get questions from all the systems from this topic, right? Not just immunology. You can get questions in GI tract, you can get questions in cardiovascular system about the chronic rejection, chronic transplant rejection, or graft versus host disease. Okay. So this is more of a transplant topic rather than immune response topic. Okay. So the first is hyperacute, that is within minutes and within 48 hours. Now the pre-existing recipient antibodies react to the donor antigen through the type 2 hypersensitivity reaction, and it activates the complement 2. Right. So it is due to the preformed, we are going to write in short about the pathogenesis here, is due to preformed antibodies. Okay. That is type 2 hypersensitivity reaction, as we saw the hypersensitivity reactions. Right, it is the inflammation. Okay, because there is also activation of the here it is hyperacute transplant rejection. So the binding of antibodies, recipient antibodies, to the cell surface of the donor antigen, which it will cause activation of complement system and Fc receptor mediated inflammation. So there is complement activation too with hyperacute transplant rejection. Right? So the C3, C4 are going to be decreased. And the features are the most important. Okay, so there is widespread thrombosis of graft vessels, arrows within the glomerulus A. Okay, so these are the thrombus formations. Okay, and ischemia and fibrinoid necrosis. So the graft must be removed. In acute, there's a week to months, but less than 6 months. Till 6 months, it can be called acute transplant rejection. Okay. So there are two types of immunity getting activated. First is the cellular. Cellular, as you know, it is through the cytotoxic T cell, CD8 plus T cells, or CD4 plus T cells activated against donor MHCs. Okay. That is type 4 because T cells are getting involved. The hyperacute was type 2 because antibodies were getting involved. In type 4, there is just T cells getting involved, no antibodies. Right? and humoral similar to hyperacute, except the antibodies develop after the transplant. There are no preformed antibodies as in hyperacute, okay, but in acute humoral there is antibodies that are being developed after the transplant takes place, okay, associated with C4D deposition, complement C4D deposition. So there is going to be CD4 plus and CD4 CD8 plus T cells. And the features are vasculitis of graft vessels with dense interstitial lymphocytic infiltrates in B. So these are the lymphocytic infiltrates in the vessel wall, right? That is the definition of vasculitis, right? That presence of lymphocytes and neutrophils in the vessel wall. So it can be prevented and reversed with immunosuppressants. Okay, so the CD4 plus and CD8 plus cells, CD8 plus T cells are the inflammatory lymphocytes, right? It is going to induce immune response, and the immune responses are occurring on the vessels wall of the graft vessels, right? So there is vasculitis of graft vessels. These are lymphocyte. T cells are lymphocytes, right? So there is a lymphocyte infiltration on the vessel wall causing vasculitis. That's how you can remember it. Now the chronic transplant rejection. Now the chronic transplant rejection, it takes place between months to years. Okay, and the CD4 plus T cells respond to recipient antigen presenting cells presenting the donor peptides, including allogenic MHC. Okay. Both cellular and humoral components are involved, that is type 2 and type 4 hypersensitivity reactions. Okay, so like if this is the APC and this is the recipient APC, it is carrying a donor antigen. Okay. And it binds to the CD4 T cell. That is recipient CD4 T cell. Okay, so that is going to activate the CD4 plus T cell, and that is going to secrete cytokines. That is going to cause inflammatory response. It has both cellular and humoral components, just like acute transplant rejection. Okay. And so the recipient T cells react and secrete cytokines, and these cytokines promote inflammatory response by proliferation of vascular smooth muscle, parenchymal atrophy, and interstitial fibrosis. It is dominated by arteriosclerosis in the C. If you can see, there's thickening of the wall, right? That is called arteriosclerosis. And organ-specific examples are chronic allograft nephropathy, bronchiolitis obliterans, accelerated atherosclerosis in heart. Okay. This is a very important point. And there's vanishing bile duct syndrome, or there is reduction in the lumen of the bile duct. Okay. There's both type 2 and type 4. Acute, hyperacute had just type 2, acute had type 4, and chronic have type 2 and type 4 as well. And in graft versus host disease, the onset varies, right? Thus, grafted immunocompetent T cells proliferate in the immunocompromised host and reject host cells with foreign proteins. Right? So there is severe organ dysfunction. So it is due to the donor T cells. Here it was recipient T cells. Right. So grafted immunocompetent T cells proliferate inside the immunocompromised host and reject the host cells with foreign proteins, so that will lead to severe organ dysfunction. It is mostly due to the HLA mismatches, most importantly HLA-A, B and DR antigens increase the risk for graft versus host disease, and it is a type 4 hypersensitivity reaction again. Okay, so that is type 4 hypersensitivity reaction because there is no antibodies involved, it is just donor T cells. And the presentation of GVHD is very unique. You can always figure it out in the questions term. It is maculopapular rash, jaundice, diarrhea and hepatosplenomegaly. And usually in bone marrow and liver transplant because they are rich in lymphocytes, right? Those bone marrow grafts and liver grafts are rich in lymphocytes. Okay, and they are potentially beneficial in bone marrow transplant for leukemia because of the graft versus tumor effect. Because the donor recipient is having the tumor, right? And if the graft has lymphocytes which can attack those tumor cells, it would be beneficial for the patient too, right? And for patients who are immunocompromised, irradiate the blood products prior to transfusion to prevent GVHD. So that is called leukoreduction, I think. Okay. So in immunocompromised patient, you have to irradiate the blood products prior to transfusion. Now the immunosuppressants part, agents that block lymphocyte activation and proliferation are called immunosuppressants. Block lymphocyte activation and proliferation, and reduces acute transplant rejection by suppressing the cellular immunity, right? Because acute transplant rejection, we saw that it is mediated by the CD4 plus and CD8 plus T cells. So for CD4 and CD8 to form, we need first of all activation and then differentiation of T cells into CD4 and CD8, right? And it is used as prophylaxis, frequently combined to achieve greater efficacy with decreased toxicity. And chronic suppression increases risk of infections and malignancy because we are suppressing the immunity, right? Okay, so let's understand this diagram. So this is the T cell receptor. It starts from here. This is the T cell receptor, right? So once the T cell receptor, once the antigen presenting cell or they bind to the T cell receptor, right, through the MHC complex, if they bind to the T cell receptor, what is happening is activating calcineurin, right? And what calcineurin does is it converts NFAT-P into NFAT, right? And NFAT is a transcription factor. Okay. Now the activated transcription factor NFAT, it activates the transcription of inflammatory cytokine genes, right? So that those cytokines can mediate the inflammation further. So NFAT is a transcription factor which activates the transcription of inflammatory cytokine genes that will mediate the inflammation further on. Right? So this is the transcription occurring. The inflammatory cytokine genes have been formed, right? And there's another mechanism through which inflammatory cytokine genes or proliferation genes are also transcribed, that is through IL-2 receptor. When the interleukin 2 binds to the IL-2 receptor, where is the IL-2 coming from? It is coming from the undifferentiated T cell, right? We saw on the interleukins part, you can still go back and see that part. Interleukin-2 is secreted from the undifferentiated Th cell, okay, before it is differentiated into Th1 and Th2. So interleukin-2 binds to the interleukin-2 receptor. Interleukin-2 receptor activates mTOR, and mTOR is a transcription factor that is going to increase the proliferation genes transcription, okay, increase the transcription of the proliferation genes, right? And for the cell to replicate, okay, or to divide, we also need purine nucleotides, right? That is how the DNA is formed. That is the base of the DNA, the nucleotides and the purine and pyrimidine bases. So purine nucleotides are formed by IMP dehydrogenase and PRPB amidotransferase, right? So both of them gives rise to the purine nucleotides, and purine nucleotides give rise to the DNA replication, right? Now let's see the drugs acting on the different stages of this process. So the first is cyclosporin, okay. So cyclosporin inhibits the calcineurin, okay. And how does it make that happen? It binds to the cyclophilin. So cyclophilin is required by calcineurin. It is required for the calcineurin formation. So cyclosporin binds to the cyclophilin and enables it to bind to the calcineurin, okay. So cyclosporin inhibits calcineurin through binding of cyclophilin, and tacrolimus binds to FKBP. And what is the form? Okay, so that is FK506 gene binding protein, okay. So that is FK506 binding protein. So tacrolimus binds to that and inhibits the formation of calcineurin. Then the glucocorticoids directly inhibits the transcription of NFK beta and inflammatory and the proliferation genes and inflammatory cytokine genes. Okay. It inhibits NFK beta, so suppresses both B and T cell function and decreases transcription of many cytokines. Okay. Okay, so glucocorticoid is inhibiting the NFK beta transcription factor. Right. Now let's talk about the daclizumab and basiliximab. So that inhibits the interleukin-2 receptor. Okay, so interleukin-2 does not bind to interleukin-2 receptor and gives rise to mTOR. And sirolimus and rapamycin. So rapamycin is a kind of sirolimus. So that binds to FKBP, that is again FK506 binding protein, and inhibits the mTOR. Okay, and this we've seen in biochemistry also, that azathioprine, and it gives rise to the antimetabolite 6-mercaptopurine, which will inhibit the PRPV amidotransferase, and mycophenolate is going to inhibit IMP dehydrogenase, that will lead to inhibition of purine synthesis, right? The cyclosporin is its mechanism as we saw, calcineurin inhibitor. It binds to cyclophilin. It blocks T cell activation by preventing the IL-2 transcription. So this is a transcription factor, that NKF beta and NFAT, is giving rise to transcription of IL-2. And that IL-2 by Th cell is going to stimulate the IL-2 receptor. It's the same IL-2. Right? And the indications are psoriasis and rheumatoid arthritis. We are going to see psoriasis at so many places in the immunosuppressant section. Okay? And its adverse effects are nephrotoxicity. This is the most important one, nephrotoxicity, and hypertension, hyperlipidemia, neurotoxicity, gingival hyperplasia, and hirsutism. Okay. And tacrolimus is FK506 binding. So it's a calcineurin inhibitor. It binds FK506 binding protein. It blocks T cell activation by preventing the IL-2 transcription. So both of them, cyclosporin and tacrolimus, inhibits the transcription of interleukin 2. Indications are immunosuppression after a solid organ transplant, and the toxicity includes similar to cyclosporin, that is nephrotoxicity, neurotoxicity, hypertension, hyperlipidemia, and all that. But there is no gingival hyperplasia or hirsutism. Right? This is unique to cyclosporin. And there's increased risk of diabetes and neurotoxicity, just like in cyclosporin. And both calcineurin inhibitors are highly nephrotoxic, so they are not given after a kidney transplant. We just give sirolimus or basiliximab after kidney transplant. That we'll see later on. Both calcineurin inhibitors are highly nephrotoxic, especially in higher doses or in patients with decreased renal function, like CKD or diabetes nephropathy, or any condition that can give rise to decreased renal function. Now, sirolimus or rapamycin, it's an mTOR inhibitor. It binds the FKBP. Tacrolimus binds FK binding protein, but they both binds FKBP, but in sirolimus the FKBP gives rise to mTOR. So sirolimus is a mTOR inhibitor. It blocks T cell activation and B cell differentiation by preventing response to IL-2. So now let's see the interleukin part. It stimulates the growth of helper T cells, cytotoxic T cells, and regulatory T cells and NK cells. Okay. Okay, I can't find the reason it inhibits B cell and differentiation too. Okay. So this might be the common pathway of both the lymphocytes, B and T lymphocytes. Okay, so that is why it is blocking both B cell and T cell differentiation. IL-2 promotes differentiation of both T cells as well as B cells. Okay. And it is indicated in kidney transplant rejection prophylaxis, specifically Sir Basil's kidney transplant. If any of you guys make it till the end of this video and you know the exact reason why it is also blocking the B cell differentiation, feel free to mention it in the comment section. Okay. And the toxicity causes pancytopenia, that is pronounced as pancytopenia. Okay. Insulin resistance, hyperlipidemia, and not nephrotoxic. Okay. Obviously this shouldn't be nephrotoxic because we are using it after kidney transplant, okay, to for prophylaxis of transplant rejection, right? So it should not be nephrotoxic at all. And it is synergistic with cyclosporin, can be used with cyclosporin, also used in drug-eluting stents. Okay. And veseleximab is a monoclonal antibody that blocks IL-2 receptor, and its toxicity gives rise to edema, hypertension, and tremor. And it's also used after kidney transplant. Okay. And also, sirolimus is insulin. It gives rise to insulin resistance and hyperlipidemia. That is why it is not given. It is contraindicated in metabolic syndrome. And the full form of mTOR is mammalian target of rapamycin. Not that you guys wanted to know that. Okay. Okay, so azathioprine, the mechanism it's anti-metabolite precursor of 6-mercaptopurine. So azathioprine gives rise to 6-mercaptopurine, which inhibits lymphocyte proliferation by blocking the nucleotide synthesis by blocking PRPB aminotransferase, right? Okay. Its indications are rheumatoid arthritis, Crohn's disease, glomerulonephritis, and other autoimmune conditions. And toxicity gives rise to pancytopenia, just like sirolimus. Okay. And 6-mercaptopurine degraded by xanthine oxidase. So toxicity increases by allopurinol, because allopurinol, as we all know, inhibits xanthine oxidase. So if xanthine oxidase is now inhibited, there would be no one to degrade the 6-mercaptopurine, right? And mycophenolate mofetil, that reversibly inhibits IMP dehydrogenase. Okay, it reversibly inhibits IMP dehydrogenase, preventing purine synthesis of B and T cells. So maybe it is reversibly inhibiting IMP dehydrogenase. So if we provide more substrate for the inosine monophosphate formation, we can reverse it, right? What is the precursor for IMP? And I think it's xanthine or guanine. Yeah, it's hypoxanthine, right? Guanine gives rise to GMP, okay, and hypoxanthine gives rise to IMP, and adenine to AMP, right? Its indications are glucocorticoid. What happened? Right. So its indications are glucocorticoid sparing agent in rheumatic disease. So if you don't want to use glucocorticoids, we can still use mycophenolate mofetil. Right. And its toxicity gives rise to GI upset, pancytopenia, hypertension, and it's less nephrotoxic and neurotoxic. But it is associated with invasive cytomegalovirus infection, okay. So every drug except cyclosporin and tacrolimus have pancytopenia, right? And except glucocorticoids, right? Every drug is having toxicity of pancytopenia. And finally, the glucocorticoids that inhibits NFK beta transcription factor. So the transcription of IL-2 is reduced. It suppresses both B and T cell function. And just like in sirolimus, we saw as it was blocking the IL-2 receptor. It was blocking mTOR through binding of FKB beta and reducing the differentiation of T and B cells both. Glucocorticoids also suppress both B and T cell function by decreasing the transcription of many cytokines, and it induces T cell apoptosis. Okay. Many autoimmune and inflammatory disorders, adrenal insufficiency, asthma, CLL, non-Hodgkin's lymphoma. Really, we all know the indications of using glucocorticoids. Okay. And its toxicity also, we've studied this toxicity since second year, right? There's Cushing syndrome, osteoporosis, hyperglycemia, and diabetes, amenorrhea, adrenocortical atrophy, peptic ulcers, psychosis, cataracts, avascular necrosis of femoral head in elderly women, and demargination of WBCs cause artificial leukocytosis. Okay. So there is artificial leukocytosis, and thus adrenal insufficiency too may develop if drug is stopped abruptly after chronic use, mostly in Addison's disease. That is called Addison's crisis. If the patient is not compliant with the glucocorticoids or is stopping glucocorticoids suddenly, they will present with Addison's crisis, which is with severe abdominal pain. Okay. And the recombinant cytokines and its clinical uses. So the first section is bone marrow stimulation, and the cytokine is erythropoietin. We all know erythropoietin stimulates the RBC production, right? And the recombinant agent is epoietin alpha, EPO analog. Okay. It's used in anemias, obviously, especially in renal failure, because in renal failure there is EPO deficiency, because EPO is secreted from kidneys. It is associated with increased risk of hypertension and thromboembolic events, and because we are increasing the viscosity of the blood by increasing the RBC mass. Right? Colony stimulating factors, that is filgrastim and ceregramostim. Right? Filgrastim is a GCSF analog, granulocyte colony stimulating factor, and ceregramostim is GM-CSF. Okay. It's used in leukopenia, obviously when we want to increase the number of leukocytes and recovery of granulocyte and monocyte counts. Right. Thrombopoietin, that is to increase the platelet secretion, platelet formation. The agent is romiplastim. It is a TPO analog, thrombopoietin analog, L-thrombopeg. Okay, romiplastim and L-thrombopeg. Think L-thrombopeg, TPO receptor agonist. Okay, so it increases the activity of thrombopoietin on thrombopoietin receptors. Right? It is used in autoimmune thrombocytopenia. It is a platelet stimulator. So for RBC, we have erythropoietin, that is epoietin alpha. For WBCs, for leukocytes, we have colony stimulating factors, that is filgrastim and ceregramostim. And for platelets, we have romiplastim and L-thrombopeg, that are thrombopoietin analogs. Right? What is the immunotherapy? Is interleukin-2 and interferons. So interleukin-2, the agent is L-leukin, is used in renal cell carcinoma and metastatic melanoma. Okay. There are very vague clinical uses. Okay. Renal cell carcinoma and metastatic melanoma. And interferon alpha is used in chronic hepatitis C, but it is not preferred much. We give mostly sofosbuvir and that other valpatasavir, something. Yeah, valpatasavir. So chronic hep C and B and renal cell carcinoma. And interferon beta is used in multiple sclerosis. And interferon gamma is used in chronic granulomatous disease, because gamma granulomatous disease. And interferon beta, we know it's used in commonly in multiple sclerosis, right? And it's a very expensive drug too. Okay, and other two drugs I would like to discuss is just one drug. That is abatacept. It inhibits CD80-86, that is B27. Okay, so that is the B7 of B cell that was going to bind to CD28 of T cell, right? So that was the secondary co-stimulatory signal, right? And also I would like to discuss post-transplant lymphoproliferative disorder. So after immunosuppression, Epstein-Barr virus affects the lymph nodes. Due to decreased T cell immunity, that is cell-mediated immunity. So the symptoms are classical Epstein-Barr virus, that is fever, night sweats, lymphadenopathy, and hepatosplenomegaly. Okay. So which are the therapeutic antibodies? Okay. So the first is autoimmune disease therapy. The first is adalimumab and infliximab. So the target is TNF alpha. So we saw TNF alpha was the acute inflammatory reactor along with IL-1 and IL-6. Right. So they are used in inflammatory bowel disease, rheumatoid arthritis, ankylosing spondylitis, and psoriasis as well. The is used in pre-treatment screening of CMV, EBV, HBV, HCV, TB, varicella zoster virus due to risk of reactivation. This should be done before starting this adalimumab and infliximab therapy. Etanercept is a decoy TNF alpha receptor and not a monoclonal antibody. Okay. Okay, and with chronic therapy of TNF alpha inhibitors, there can be neutralizing antibodies formed in the body. Okay, so those neutralizing antibody will decrease the clinical response of TNF alpha inhibitors. That can happen. Okay, and eculizumab is for complement protein C5 for PNH, especially for PNH, and it is associated with increased risk of meningococcal infection. Guselkumab for IL-23 for psoriasis, ixekizumab and secukinumab for IL-17A for psoriasis, psoriatic arthritis, natalizumab for alpha-4 integrin for multiple sclerosis and Crohn's disease, and alpha-4 integrin is used in WBC. Alpha-4 integrin is used in WBC adhesion, and there's risk of PML in patients with JC virus. Okay. What was PML? Promyelocytic leukemia or something like that? No. Its progressive multifocal leukoencephalopathy is an opportunistic infection of the central nervous system caused by the human polyoma virus, that is JC virus. Yeah. Characterized by focal demyelination. Alright. And ustekinumab is for IL-12 and IL-23. Clinical use is psoriasis and psoriatic arthritis. Vedolizumab is for alpha-4 integrin again for inflammatory bowel disease. It's a gut-specific anti-integrin, okay, preventing migration of leukocytes to the gastrointestinal tract. Denosumab is a rank ligand inhibitor. Okay. So as we saw earlier, that rank ligand is used in osteoclast. Okay, so and osteoclast is known for increasing the bone resorption, right? So there is going to be demineralization in the bones. Okay, so denosumab inhibits the rank ligand. Okay, it is used in osteoporosis, and it inhibits osteoclast maturation, mimics osteoprontogerin. So naturally in the body, osteoprontogerin inhibits the binding of rank to rank ligand, inhibiting the activity of osteoclasts. And denosumab helps make dense bones. Emicizumab is a factor 9 and factor 10 target and is used in hemophilia A, It is bispecific, mimics factor 8. Omalizumab is for IgE, refractory allergic asthma, and prevents IgE binding to Fc epsilon RI. Okay. Just remember that it is used in refractory allergic asthma. And Pelevizumab is for RSVF protein. It is used in RSV prophylaxis for high-risk infants. Yes, mostly used in the US, not in developing nations like India. Okay, and a few, 2 or 3 other drugs like Epiciximab. Right. Epiciximab binds to glycoprotein 2B and 3A. Right. I'll show you how I remembered that. Glycoprotein 2B and 3A. So 2 into 3 is 6. Okay, and this is 6. Right. It prevents ischemia events in patients undergoing percutaneous intervention following myocardial infarction or atherosclerotic disease. Okay. Prevents ischemia events in PCI patients undergoing PCI. Okay. And bevacizumab it targets VEGF, that is vascular endothelial growth factor, used in colorectal carcinoma, renal cell carcinoma, non-small cell lung carcinoma, okay, macular degeneration, diabetic retinopathy, but the proliferative type, and macular edema. So which were the drugs used in renal cell carcinoma? The bevacizumab. Then there was interleukin 2 recombinant agent, that is aldesleukin, right, and interferon alpha, right. Okay, so yeah, just one more drug, that is LM2zumab, is used in CLL and multiple sclerosis. Okay, so we are done with immunology part.